crop production technologies. On behalf of the organizing committee of Nsatrs 2024, it's my great privilege to introduce and welcome such a wonderful academic in the field of agriculture, Dr. Nissala Vidanapathirana. With a wealth of experience in on behalf of the organizing committee of Nsatrs 2024, it's my great privilege to introduce and welcome such a wonderful academic in the field of agriculture, Dr. Nissala Vidanapathirana. With a wealth of experience both teaching and research, she consistently demonstrated excellence dedication through her career. Currently, she acts as the Department of Technology Senior Lecturer in UCIRS. We are honored to have such a great person in our institute. So, dear madam, it's over to conduct the session.
Thank you, Mr. Buddhika. We are going to start our first technical session, crop production and protection technologies. First of all, I would like to welcome and to our session, our panel member, Mr. H. Rohanadira, who is the scientific assistant in our UCIRS. And for throughout the session, he is also using his expertise to success of the session. Okay, then first shall we start? That one is a case study on consumption pattern and awareness of underutilized foods among selected undergraduates of Faculty of Agriculture, University of Jaffna, Sri Lanka. The presenter is KPL Arosha, and also the corresponding author is KPL Arosha. I would like to invite Miss KPL Arosha to present her valuable research ideas to the panel, and would like to welcome.
Wishing you all a very good day, and I'm Arosha from Faculty of Agriculture, University of Jaffna. Here I'm going to discuss about a case study which I have done on consumption pattern and awareness of underutilized fruits among selected undergraduates of Faculty of Agriculture, University of Jaffna. Let's move to the introduction part. Sri Lanka is a natural hotspot of underutilized wild edible plants. There are around 240 wild relative species can be found in Sri Lanka. And there are several characteristics of underutilized vegetables and crops. To find out the consumption pattern, knowledge, attitude over selected the underutilized fruits by selected undergraduate University of Jaffna is the objective of this case study. A self-administered online survey was conducted among selected undergraduates, and data was analyzed using MS Excel and IBM SPSS software. And these are the selected underutilized fruits. 44% of the respondents were first years, and 70% of the respondents were females. 32% of the respondents were below to 22 years old category, and majority of the expend more than 6,000 rupees as their monthly expenditure. Most of the students were from Northwestern Province, and almost all students were obtaining their meal from the canteen. When they are choosing food, the major criteria for selection of food is the price, and 66% of the students are aware about the underutilized fruits as well as vegetables, and 34% of the students are not aware about the underutilized fruits and vegetables. And from that, almost all are from first year. 46% of the respondents are having average awareness on nutrient profile of underutilized foods. As per the barrier for underutilized food consumption, majority of the students have stated that non-availability of the underutilized fruit is the barrier. And as per the remedy, half of the respondents have stated that introduction of underutilized fruits and vegetable subsidies to the premises will be a great remedy to enhance daily consumption of underutilized fruits. And half of the population are having either one fruit per week. And 44% of the students are willing to have underutilized fruit as fruit juice. Daily underutilized food consumption of undergraduate students are at very lower value, and Jambu and Veralu claim to be the commonly consumed underutilized fruit. Non-availability is the biggest barrier to underutilized fruit consumption, and introduction of underutilized fruit subsidy for university students will be the remedy that respondents think may help to enhance the underutilized fruit consumption. Though students' choice of food selection depend on price, introduction of cheaper sources of underutilized fruits to the university premises can enhance the underutilized fruit consumption. And these are my references, and thank you.
Thank you very much, Ms. Arosha, for your nice presentation. And now the presentation is open for the discussion.
Okay, Mr. Arosha, hope you are joining with us.
Excuse me, madam, actually I can't hear you. But...
Mr. Arosha.
Can you hear us? Please unmute your mic, please.
Yes, madam. Now okay.
Okay, Mr. Arosha.
Good morning, madam. Yes.
Yeah, good morning, Mr. Arosha. Yeah, thanks for sharing that your valuable findings.
Can you please open your, can you please show your picture to us? Switch in our video.
Yes, madam.
Okay.
Okay, thank you, Mr. Arosha, for sharing that your valuable findings on this subject. Few things to clarify that it's very appreciated because that very minimal influence and also consideration to do this kind of research on in that underutilized fruits in this country, because there are underutilized, but the scientists are not going for such kind of great research just to make a findings and stimulate for the consumption in future as well. So in here, how many fruit varieties have you considered for this?
Actually, I can't hear you well. Am I audible?
Yeah, yes, you are okay. Can you hear me now?
Yes. Okay.
Okay. My question was that how many underutilized fruit varieties you are engaged with this research?
Yes, I have used 40 underutilized fruits.
40 varieties.
Zero.
What are the basis of this nominations? why you have selected 40 numbers? What is the basis?
Because I have searched 2-3 publications, as I mentioned in the references, they do used 40, likely they have used 30 to 40, like. So I have combined all the paper together.
Okay, thank you. Then, as per your findings, what is your suggestions? Because we have to enhance that the consumption of this kind of underutilized fruits. No, what are your suggestions for that future consumers? What are the things you can expect and you can suggest for that increase that the consumption future?
So I have used that Jaffna University because that is too far from the middle of the country. So I think all students are not having much good nutrition, but and also half from that university, Sinhala people, half from the Tamil. I think those Sinhala people also living in those underutilized crop that high areas, they too are not willing to eat those fruits because they don't have those facilities in that university premises. So I think they are having like Helabujun and other places. No? So we can introduce those underutilized fruits. As I have studied, most of the students willing to have that fruit juice. So I think we can integrate those fruits as fruit juice, or we can just use reduce beverages like, so we can just enhance the consumption.
Okay, thank you. Congratulate and hope that your findings will encourage that the consumption of this kind of underutilized in at least at your university. Okay, thank you very much, Ms. Arosha.
Okay, one comments, very few one. In your conclusion, you mentioned it's an unavailability of underutilized fruits. Okay, that non-availability is only for the Jaffna University undergraduates. Okay, therefore your conclusion should be narrowed down to your students, and that you have to mention that's non-availability for that particular students. Okay, otherwise that your conclusion will go for the whole country. That should be that is not a correct conclusion. Okay.
Yes.
Okay.
Yeah, madam. I am doing a survey for all the universities too. So I will conclude all the university undergraduate.
Yes, that's good. Okay. On
Yeah,
that
madam.
remarks,
Thank you for your suggestion.
yeah, we will thank for you, and you can leave. Thank you very much. Okay, then we will start the second presentation. That research is effect of metribuzin glyphosate application and manual weeding on growth, yield, and cost for weeding in production of soybean. The presenters are Ranavira Basuri and Kumara Singha. The corresponding author is Mr. Kumara Singha, and presenter is Ms. BSS Basuriya. We will invite Ms. Basuriya to present her valuable findings with us.
Welcome you all for this insightful presentation. Today we are excited to delve into an important study that examines the effect of metribuzin glyphosate application and manual weeding on growth, yield, and cost for weeding in the production of soybean. I am Sachini Basuria and I will be presenting our research findings with significant contribution from Senior Lecturer Mr. H.K. Ms. Kumar Singha and Ms. Ranavira. Are the major topics which are presentation. So let's introduce the background research. Soybean, a legume rich in oil and protein, is essential for various food, animal feed, and other industries, with global production reaching 388 million tons in 2023. Sri Lanka produced 3.84 kilotons in 2022, mainly from dry and intermediate zones in Sri Lanka. Weeds are the major biological constraint to crop yields, causing 26 to 84% losses in globally. An effective control method, especially herbicides, are crucial for optimizing productivity and resource efficiency, as uncontrolled weed can reduce the soybean yield by up to 1% per day after the V2 growth stage. Let's move on to the specific problem faced by this industry. Weeds are a major problem in soybean farming, causing yield reduction between 26 to 54%. One study reported a 4 to 5% reduction in soybean yield due to the weeds. To control weed, farmers use both manual and chemical weeding methods. Manual weeding requires 30 labors per hectare per cycle, which means 60 labors per 2 cycles per hectare, with the total labor cost around 1,20,000 rupees or more than that. Due to the low availability of labor and high cost, farmers are increasingly turning into the chemical weeding. This study investigated the efficiency of the pre-emergence and post-emergent herbicide, primarily metribuzin glyphosate, and manual weeding on weed management in soybean cultivation with the focus on their impact on growth, yield, and associated cost for weeding. The main objective of this study is to evaluate the impact of metribuzin, glyphosate, and manual weeding on growth, yield, and cost of weeding of soybean. Next, we explore how this study was carried out. The study was carried out at the Faculty of Agriculture, University of Pune, and we examined the various weeding methods, including manual weeding, metribuzin, and glyphosate application. We measured the growth and yield parameters, and the data were analyzed by using one-way ANOVA, and the means were separated by using Dunner test at 5% probability. This experiment was conducted using completely randomized design with 4 replicates, and this is the way of field arrangement. Treatments were applied as shown in this table. To collect the data, we utilized a sample of 12 predetermined plants. This is the irrigation schedule we followed from seed sowing to the harvest. The fertilizers were applied according to the recommendations of the Department of Agriculture. This is how we practice thinning. These pictures summarize our research from beginning to the harvesting. With our methodologies in place, let's reveal what we discovered through our research. The study found metribuzin significantly reduced weed count after first weeding at 14 days after seed sowing, with T2 and T3 outperforming T1, but no significant effect were observed after second weeding at 35 days after seed sowing. There was no significant difference among the treatment for the plant height. Sharma and others found that metribuzin reduced the plant height and growth rate in the lentils plant. And one research found that increased the glyphosate application decreased the soybean plant height. There was no significant difference among the treatment for the number of leaves per plant. Number of shoots per plant at 80 days after seed sowing were not significantly among the treatment. The study found no significant effect on the 50% flowering time. It takes only 35 to 36 days for all treatment. Research shows that glyphosate prolongs the flowering due to the physiological impact, and while metribuzin generally causes a less delay but may induce stress-related slight delays. Number of pods per plant at 80 days after seed sowing were not significant among the treatment. The pot filling percentage was calculated as 3-seeded, 2-seeded, 1-seeded, and infill. According to the results, there was no significant difference in the all treatment compared to the T1 for the percentage of pot filling. The results showed that there was no significant difference for the seed weight compared to T1. Based on the prevailing market for chemicals and labor input during the study period, the cost analysis revealed that manual weeding incurred the highest expenses for weeding, whereas the application of glyphosate for 2 weeding cycles required least cost approximately 26,000 rupees per hectare. According to the results obtained by this research, it can be concluded that metribuzin and glyphosate did not significantly affect the growth and yield of soybean. The chemical weeding was more cost-effective than manual weeding in soybean cultivation. Among the treatment methods, irrigation is the most cost-effective for both weeding cycle in soybean cultivation. These are the references of this study. Thank you very much for your attention.
Thank you very much for your nice presentation.
Basudri, I hope that you can hear us. Basudri, can you hear us?
Yes, sir. I can hear you.
Okay, nice presentation. Few clarification that
Thank you.
we have selected 2 chemicals that use glyphosate, right? like
Yeah.
two chemicals. But
Sorry,
you
sir.
select only two chemicals for this.
Actually, glyphosate is post-emergence herbicide and metribuzin is a pre-emergence herbicide. Using post-emergence herbicide by followed by the pre-emergence herbicide is most effective then. So that's why I use both two chemicals for weeding because it was very effective controlling the weed.
Thank you. And today, the farm cultivation herbicide for the weed control system. Okay. And also, you have
Yeah.
conducted this research in net house, no? In the net house condition.
Sorry, sir. I can't clearly hear you.
Have you conducted this research under net house conditions?
Yes, sir. I conducted inside the protocol.
Okay, that's good. Then, how you have introduced the weeds in your pit floors?
I use normal field soil. I think with them weeds and others weed seeds come from that with the soil because I use soils in field conditions.
Okay, it means that you didn't introduce the weeds that natural soil having that seeds.
No, sir.
So,
Yeah. Yeah.
okay, that's good. Then your finding reveal that the glyphosate and metribuzin have any significant difference when controlling that and also
Yes, sir.
that the your parameters, ne? So,
Yes, sir.
what is your final message to the farming community in this country?
Most of farmers are challenged due to the weed. So to control the weed, so I recommend using weedicide, so herbicide, especially glyphosate for both two weeding cycle. It is most cost effective than manual weeding. Because labor cost is very high at present situation and it is very time consuming. Therefore, I recommend use of weedicide rather than using manual weeding.
Okay, thank you, Ms. Basri and team. It's a nice presentation and nice findings for your research has conducted. Thank you very much.
Thank you. Thank you very much, Madam and Sir.
Yeah. Thank you very much, Basuria. And we will move to the next research findings. That is, study the impact of gibberellic acid inhibition by foliar application of cyperus on growth and yield of curry chili cultivated in shaded protected house. The presenter is Miss Vijay Sundara, and we will invite Miss Vijay Sundara to present her valuable findings with us.
Hello everyone, I am Ishika Vijayasundara. Today I will be presenting our study on the impact of inhibiting gibberellic acid through foliar application of paclobutrazol on the growth and yield of curry chili plants cultivated in a shaded protected house. The study was conducted by HKMS Kumarasinghe and WMSI. Our study focused on the use of plant growth retardants to manage chili plants. Plant retardants such as paclobutrazol play a crucial role in modifying plant growth patterns, ensuring optimum plant structure and enhancing overall yield under protected cultivation conditions. To set the stage for our discussion, let's move on to the research problem. The adverse effects of high temperatures in greenhouse cultivation results in pests, diseases, and pollen inviability. However, we recognize the potential of shade as a solution. Shade excessive shading poses its own problems, including stem elongation and fruit thickening due to gibberellin production. Paclobutrazol is a synthetic triazole with the power to inhibit vegetative growth by targeting gibberellin biosynthesis and abscisic acid catabolism. The primary objective of this research was to develop a protocol to cultivate curry chili under 50% shade in low country with minimum effect on growth and reproductive parameters. This experiment was conducted at Faculty of Agriculture, University of Brunei, arranging in a randomized complete block design with 6 treatments and 5 blocks. 6 different concentrations of paclobutrazol were sprayed to the foliage at 14 days interval. Data collection was done at 14 days interval as growth data and reproductive data. Results and discussion. Plant height was significantly affected by the rate of paclobutrazol. Lower doses of paclobutrazol resulted in a smaller reduction in plant height. This effect is due to gibberellin synthesis inhibition, which suppresses stem elongation, and its impact on other plant hormones such as abscisic acid and cytokinins, which have opposing effects on stem growth. The application of paclobutrazol resulted in a significant reduction in internodal length attributed to its suppression of gibberellin synthesis, consequently compacting the same number of leaves and internodes into a shorter length. Significant variations in stem diameter were noted across different concentrations due to enhanced secondary xylem development and reduced sclerenchyma bundle caps. The study found that the rate of paclobutrazol application significantly reduced the number of leaves per plant. Study found that in several plant species, onset of senescence was considerably delayed in paclobutrazol-treated leaves due to influence of paclobutrazol on the endogenous cytokinin content. Moving on to the reproductive parameters, the results illustrated that the number of fruits per plant was significantly influenced under increasing concentrations of paclobutrazol. The reduction of plant height with paclobutrazol indirectly reduces leaf area, which may reduce photosynthetic surface area and thereby reduce the whole plant photosynthesis. Weight of single fruit also reduced with all applied paclobutrazol doses. Reduced growth and number of leaves have ultimately reduced the photosynthesis, leading to low weight fruits. Due to its effects on plant height, paclobutrazol also significantly impacted on fruit length. By reducing gibberellin levels, it can cause the cells to divide more rapidly, however it limits cell elongation. Consequently, the expansion of fruits is reduced, leading to smaller fruits. Compared to the control, the treated plant showed a decrease in trend in fruit diameter due to reduced cell expansion caused by gibberellin inhibition. In conclusion, the application of paclobutrazol led to a marked reduction in vegetative growth of plants across all tested doses. Additionally, it significantly affected the number of fruits per plant and length of individual fruits. Among the various concentrations tested, the treatment, which involved a 30 ppm concentration of paclobutrazol, consistently demonstrated superior results compared to other concentrations. Our research indicated that successful cultivation of curry chili in protected houses within the low country wet zone of Sri Lanka can be achieved with the 30 ppm foliar spray of paclobutrazol applied bi-weekly. These findings provide a practical and effective approach for farmers in this region. These are the references. And thank you all for listening.
Okay, thank you very much, Ms. Vijay Sundara, for your nice presentation. And now presentation is open for the discussion. I have one small clarification that for your research, did you conduct cost benefit analysis for that? Cost analysis? That you are using paclobutrazol for that, foliar application. For that, we are going to introduce that for the farming community. Okay,
Yes.
then did you conduct a cost analysis for that?
No, but the cost for paclobutrazol is very low and use very low amount. So no need.
Okay, in that remarks, you can introduce that for the farming community also.
Yeah.
Yeah. Okay.
Okay, Ms. Vijayasundara, two
Yes.
clarification from my side. Did you analyze that glyceric acid concentration, mean that traces concentrations of that your particular samples, mean that chemical analysis of glyceric acid?
No, sir.
No, very important because then you can clearly discuss in that fact. If you have analyzes that when we are increasing the concentration of that particular paclobutrazol chemical, that the glyceric acid form or existing amount is reducing. No, as
Yes.
your result shows in that manner. Okay, it's better if you have enough samples, you can do it. Doesn't matter. But for problem is that when you are going for the your conclusions, when increasing the concentration of this particular chemical, the yield is reducing.
Yeah.
What is the combination correlation between these two?
Yeah, these showed that decreasing in the vegetative parameters, but with reduction in reproductive parameters as well. But when compared to the treatment T2 with 30 ppm concentration, it has a marked reduction in vegetative parameters, especially the plant height and internode length, and with no significant effect on reproductive parameters, and no very few, very less reduction in the number of fruit content. So we found that using T2, the 30 ppm concentration was very effective rather than going for very high concentrations.
It means that when comparing that your control, the lowest concentration of this chemical is no insignificant difference between the yield parameters, no?
Yeah.
Okay.
It's quite better than going for higher concentration.
Yeah, that's great. Yeah. And also, I think we could utilize this kind of chemical for reduce that the plant height or form a bushy type in plant tissue culture as well. It's a good chemical, more or less that scientists utilize for this kind of purpose. So, do you have any recommendation for the farming community to utilize this kind of chemicals for reduce that the plant height and internal length as well?
Yes, paclobutrazol is using in the industry for early off-season flowering, and especially in some curry chili varieties for bushy type, taking bushy type plants, especially ornamental peppers. And we can use them for our curry chili as well for reducing the growth, especially the plant height when cultivating under shade conditions, because using a shade will more effective than going for misters or foggers. For temperature control, it is low cost, and we can use paclobutrazol for the problem arising when using shade to reduce the plant height. So I think using paclobutrazol, it is actually cost is also low, and also we can use it as a soil drench as well. So we can use it with the fertigation systems. It is easy to apply as well, so we can use paclobutrazol in the curry chili cultivations in Sri Lanka under protected conditions.
Okay, thank you very much. You are sharing your new findings with our symposium as well, and also wish for your future endeavors as well. Thank you.
Okay, thank you, sir.
Okay, thank you, Ms. Vijayasundara. And we will move to the next research presentation, that is impact of smart fertilizers as a strategy for better crop production in tropical agriculture. The presenter is Ms. De Silva, and I will invite Ms. Silva to present her valuable findings with us.
Good morning to you all. I am H.T. De Silva from Department of Agricultural Chemistry, Faculty of Agriculture, University of Jaffna. Today I am here to present a review on the topic Impacts of Smart Fertilizers as a Strategy for Better Crop Production in Tropical Agriculture. First, let's move into the introduction. As you all know, high population growth across the developing world means there are more mouths to feed, but also smaller farmer sites to produce foods. Food security is one of the major concerns for all developing countries of the world, and even though we had attained the highest food production with the use of new technologies, we may not able to feed the burgeoning population adequately in coming years due to stagnant crop productivity. Therefore, farmers want to cultivate more lands, or they want to practice the technology-efficient methods to increase the crop production in quantitatively as well as qualitatively. Present agriculture is generally chemically intensive, where using more doses of chemicals or nutrient management to get maximum production per unit area without caring about natural resources and ecosystems. In present agriculture, fertilizers contributes to the tune of 50% of the agricultural production, but higher doses of fertilizers does not guarantee to improve crop yield, but it leads to several problems like degradation of soil and pollution of surface and underground resources. In this context, formulation of smart fertilizers is a promising, rapidly evolving field of interdisciplinary research that has potential to revolutionize food systems by enhancing nutrient use efficiency of the crop fields for better crop production. Based on these facts, this study was conducted to review on the impacts of smart fertilizers as a strategy for better crop production in tropical agriculture. The study was carried out as a non-systematic review study using online databases, ResearchGate and Google Scholar by combination of smart fertilizers, tropical agriculture, impacts of smart fertilizers, and nutrient management as keywords. As per finding, smart fertilizers are a new class of fertilizers that have been developed to address the sustainability challenges associated with traditional fertilizers. These fertilizers are designed to release nutrients in a controlled manner, reducing nutrient loss and increasing crop yields. Based on the previous literature reviews, the smart fertilizers formulation include nano fertilizers, flow of controlled release fertilizers, polymers, biodegradable polymers, stabilized fertilizers, bioformulation fertilizers, and use of harvested residues. This diagram clearly indicates the positive impacts of the smart fertilizers. The purpose of development indeed smart fertilizer considers as the creation of a recovering single or multi-component fertilizer that adapts to the timing of nutrient release to the plant nutrient demand, enhancing the agronomic yields and reducing the environmental impact at sustainable cost when compared to conventional fertilizers. By enhancing nutrient use efficiency and minimizing nutrient losses through leaching and runoff, smart fertilizers contribute to tropical agriculture practices by reducing environmental pollution and conserving natural resources. Smart fertilizers actively engage in the enhancement of soil health, fostering a symbiotic relationship between agricultural inputs and the intricate microbial communities within the soil matrix, fostering crop resilience to climate variability and reducing the carbon footprint of agriculture. While smart fertilizers offer promising avenues for agriculture, their implementation faces several intricate challenges and limitations. Those include high initial cost associated with technology acquisition and development, ongoing maintenance and calibration requirements, regulatory barriers, pose additional hurdles to commercialization and market uptake, research gaps regarding the long-term environmental impacts, interaction with soil microorganisms, effect on ecosystem dynamics necessitate further research and risk assessment, phototoxicity, reactivity, and variance of smart fertilizers, and finally, exposure to xenobiotics during their application. As conclusion, the advances in the application of improved technology such as nanotechnology, biotechnology, etc. has the potential to facilitate improved nutrient management and use efficiency in agroecosystems. Smart fertilizers based on slow or controlled release fertilizers, bioformulation, nanofertilizers, sensors, etc. have been shown in improved crop yield, soil productivity, and lower nutrient loss compared with conventional fertilizers. Smart fertilizers offer benefits but also come with challenges and limitations, and so it's important to find smart solutions and use scientific methods to deal with these challenges for better farming. As future direction, I would like to mention the future research should be continuing to explore and evaluate the agronomic and environmental performance of various smart fertilizers and interaction of smart fertilizers with the environment and potential effects of human health must be explored in detail before spreading the smart fertilizers at a commercial scale. These are some references I used in this study. Thank you so much for your consideration.
Okay, thank you very much, Ms. Ila, for sharing with us your valuable findings. And now the presentation is open for the discussion.
Yeah, thank you, Ms. De Silva. One clarification that, as you mentioned, those are the review articles, no?
Yes.
Yeah, okay. Based on that, the review articles are considering that all tropical agriculture, tropical country agriculture systems, no? So then when you are discussing that the barriers and limitations in that previous slide of the last, so you have mentioned few barriers, but some barriers are not tally with that this country. So what could be your suggestions? You know that the barriers are existing now. We have to go beyond the barriers to achieve the target. So what could be your recommendation to introduce this kind of a smart fertilizer systems to this country?
Yes, sir. First, as a suggestion, I want to mention first of all, we should introduce that economically feasible smart fertilizers for farmers. First of all, we want to do that. Then that agricultural graduates, we want to go for the more researchers regarding economic feasibility as well as regarding the environmental sustainability also. Because based on the previous literature use, they mentioned some smart fertilizers cause to the environmental pollution because those some smart fertilizers combine inorganic items also. Because of that, we want to introduce economically feasible and environmental sustainable smart fertilizers for the farmers. And first, then after the introducing, we want to tell we want to go on the research for the that what is the efficiency of these smart fertilizers. Then only we can go for the commercialization of the smart fertilizers in the farm scale.
Yes, I appreciate that your answers, and also I think we think that we have to go as a holistic approach for this, no?
Yes.
As you said that all the researchers get together and do some separate researchers on this each and every smart fertilizer, and also the second part is that the farmer community should be aware on this very vastly.
Yes.
Not it's going to fail. Whatever we found through the researchers, definitely they are going not to be implemented in future in this soil. So thank you very much for your contribution.
Okay, and I have a clarification about smart fertilizers. Okay, you
Yes.
are doing a review research about smart fertilizers. Are there any clear cut to distinguish fertilizers and smart fertilizers? How you distinguish that smart fertilizers from other fertilizers?
Yes, madam. Yeah, smart fertilizers that using improved technology and smart fertilizers. That if we add more fertilizers with smart fertilizers, less amount of smart fertilizers needed to give that full amount of fertilizers, because smart fertilizers mainly focus on the slow releasing patterns and high nutrient efficiency when compared with normal fertilizers. This is the main difference between fertilizers and smart fertilizers.
Okay. Okay. Thank you. Thank you very much, Ms. Silva.
Thank you so much, madam.
Your findings. Okay. In these remarks, we will move to the next research findings. The presentation about study on suitability of different techniques for the production of organic liquid fertilizer from giant water fern and mosquito fern. The presenter is Ms. Tissera, and we will invite Ms. Tissera to share with us your valuable findings.
I am I.J. Tissera, Department of Biosystem Technology, Faculty of Technology, University of Jaffna. My research is study on suitability of different techniques for the productions of organic liquid fertilizer from giant water ferns, Salvina molesta, and mosquito fern, Azolla pinnata. These are my content. Introduction. Fertilizer. Fertilizer can be divided into the organic and inorganic fertilizer, which are supply essential plant nutrition for the plant growth. Excessive use of inorganic fertilizer make adverse impact on human health and environmental problems. Furthermore, inorganic fertilizer are highly costly than organic fertilizer, but organic fertilizer contribute to sustainable agricultural practices. Salvinia molesta. Salvinia molesta is a free-floating fern. In 1939, Salvinia molesta was introduced in our country for research purpose by Department of Botany, University of Colombo. Salvinia molesta has rapid growth rate and it was categorized under the invasive aquatic plant. So, nowadays, Salvinia molesta used for the phytoremediation, make biofuel and organic fertilizer. Azolla pinnata is a free-floating aquatic fern. It has relationship between nitrogen-fixing blue-green algae and nowadays it can be used as a biofertilizer, phytoremediation agent and as an animal feed. Problem statement. Excessive use of chemical fertilizer will make adverse impact on human health and environmental problem. Nowadays, availability of organic fertilizer is very limited in our local market. Due to that case, very important to find alternative organic source for nutrition and fertilizer product in our country. Furthermore, Salvinia molesta creates serious economic, social and environmental problem for our country. Justification. This study was aimed to analyze nutrition contents of organic liquid fertilizer prepared by utilizing rapidly growing 2 aquatic plant under the 5 liquid extraction method. Introduce low-cost organic liquid fertilizer and find the solution for the current organic fertilizer problem in our country. Objectives. Aim to produce organic liquid fertilizer by utilizing Salvinia molesta and Azolla pinnata. Specific objectives are to extract Salvinia molesta and Azolla pinnata by using 5 different liquid extraction methods and to assess Salvinia molesta and Azolla pinnata liquid fertilizer nutritional content. Materials and method. Experimental location. The experiment was conducted in the soil division at the Fruit Crop Research and Development Institute in Sri Lanka. Experimental design is a complete randomized design. Under the one-dry method, first of all, selected plant species were washed with deionized water and those were put in oven under the 60 degrees Celsius for 72 hours until dried. Finally, dried 500 milligrams of oven-dried sample mixed with 200 milliliters of double distilled water. Finally, that mixture boiled under the 15 minutes under the 100 degrees Celsius using water bath and it was filtered by using 1 pore size filter paper. Grinding method. 100 grams of selected Salvinia and Azolla mixed with 30 milliliters of deionized water. Then after, blend with both species separately and extract was taken by using muslin cloth. Finally, extract was filtered by using 1 pore size filter paper. Under these 5 extraction method have 3 digestion method. Those are the anaerobic digestion method, aerobic digestion method by using aerator and trichoderma digestion method. Under these 3 digestion method, 200 grams of both plant species were cut into the small pieces and those were put into the net bag separately. Then 2 liters of deionized water filled into the clean bucket. But under the trichoderma digestion method, trichoderma hasianum 10 grams was added into the net bag and tied it well. And it was dipped into the water bucket. Then after, aerator was connected except anaerobic digestion method. Finally, all 3 buckets were covered with the polythene. Then liquid samples were got in order to 7th, 14th and 21st days to check chemical parameters. Under the chemical parameter analysis, pH value and EC value was analyzed by using pH meter and EC meter. Liquid Azolla and Salvinia sample was digested before analyzed nutritional elements. Sample digest by using advanced microwave digestion system to convert complex elements into simple elements that easy for identify target nutritional elements. After digest the sample, determine potassium and calcium by using flame photometer. Phosphorus can be determined by using spectrophotometer. Zinc and magnesium calculated by using atomic absorption spectrophotometer. Before determining the nitrogen level, sample need to allow digestion process and and distillation process. After this digestion and distillation process, we can determine nitrogen percentage using this calculation. Finally, we can see comparing both salvinia and azolla nutrition content. Highest nitrogen content was determined in the oven dry method. Highest phosphorus content was determined in the trichoderma digestion method. Highest calcium, potassium, magnesium, zinc content was determined in the grinding method. Optimum pH and EC level can be shown in the grinding method. Conclusion, analysis was done by using statistical analysis system SAS in 9.2 version. From the result of the present investigation, there was no any special nutrition extraction done under any method. However, grinding method performed well in nutritional extraction both salvinia and azolla. In addition, overall comparisons of nutrition contents of extraction obtained from the both species revealed that azolla has higher nutrition content than salvinia, excluding magnesium. Future suggestions. Further studies are needed to confirm whether the product contains any toxic elements, have or not. Can be extended anaerobic method time duration for nearly 6 months to get liquid extract. Can be mixed both salvinia and azolla using different ratio to produce fertilizer combination for the plant growth. These are my reference. Thank you all.
Okay. Thank you, Ms. Tisera, for your nice presentation. Now the presentation is open for the discussion.
Okay, Mr. Sarah, it's very impressive findings you have gained throughout your research. One clarification I need from you. Have you analyzed the cost of production of these 5 methods?
No, sir.
No. It's very valuable if you, because sometimes that the digestion system could make some lower cost, while that grinding and other methods could make some high cost. So when you are recommending something, it's very important to check what is the actual cost we have to bear for this. Okay, then I think you have that fact, it's better to reanalyze it. And good luck. Thank you very much.
Thank you so much.
I have one clarification that in your application, that means you are producing liquid fertilizer using salvinia and azolla. Are you combine these two together, or you are doing a separated? You separated those things.
Yeah, so I am check nutrition analysis, but the 2 species are separately. Because salvinia has no any nutrition content, but already azolla have high nitrogen content. That's why I mix both species together.
Okay. Thank you very much, Ms. Tisera.
Thank you so much, madam.
Okay, in that remarks, we will move to the next presentation. That is the effect of foliar application of fish amino acid on growth and yield of tomato cultivar Padma. The presenter is Miss Nishanti, and I will invite Miss Nishanti to present her valuable finding with us.
Good morning, everyone. Welcome to my presentation. As you can see on the screen, our topic is the effect of foliar application of this amino acid on growth and yield of tomato cultivar Padma. This presentation is structured as follows. Introduction with justification, objective, methodology, research and discussion, conclusion, recommendation, and reference. Let me start by saying a few words about my research background. Tomato is an important crop plant cultivated all over the world. Inorganic fertilizer have been used as primary means of engaging soil fertility. But continued and over-application increase ecosystem toxicity. But nowadays, expensive and not available in market. So we need to find alternative source. Pistonic is such a potential organic fertilizer source while keeping during the early or vegetative stage. Therefore, this research was targeted on evaluating the effect of histonic as a potential organic fertilizer. Objective, general objective, to find out the combined effect of histonic and inorganic fertilizer application on tomato. Specific objective, to evaluate the combustion of histonic and to study the effect of histonic and inorganic fertilizer on growth and rate of tomato, and to study the effect of histonic and inorganic fertilizer on physical and chemical parameters of soil. Our methodological approaches, first of all, preparation of histonic. We would use fish waste and over-dried banana 1:1 ratio by weight. Next, experimental design. The experiment was carried out as a field pot experiment. Next, fertilizer treatment. Then, histonic was applied as a foliar spray 1 week interval according to the treatment. Then, plant growth parameters were measured 45 days after transplanting. Then, measurement of soil, physical and chemical parameters. Finally, all the data was analyzed using ANOVA method. Next, results and discussion. Let's see our results and discussion through the objective. This table shows our manual preparation of histonic combustion. Total nitrogen, phosphorus, potassium, pH, and EC. Next, to evaluate the growth performance of tomato crop in each treatment at 45 days after transplanting. Plant growth parameters are plant height, yield area index, number of branches, flowable pattern. Yield parameters are flower per plant, fruit per plant. Here, for example, this chart shows plant height performance of tomato crop 45 days after transplanting. Here, the application of 1% of histonic with 50% of inorganic fertilizer has significant increase in the growth and yield parameters compared to the 100% inorganic fertilizer. Next, the soil attribute at the initial stage of experiment. Here, at the harvesting stage, the pH wasn't significantly different among the any treatment. Here, soil application of histonic treatment now significantly higher phosphorus, potassium, nitrogen content at harvesting compared to the initial stage. Next, conclusion. The application of 1% of histonic with 50% of inorganic fertilizer has significant increase in plant vegetative growth compared to the 100% inorganic fertilizer. The application of 1% histonic with 50% of inorganic fertilizer had significant increase of number of flowers and plants per plant compared to the 100% inorganic fertilizer. This suggests application of nutrition into soil and foliar growth simultaneously will significantly increase the nutrition absorption capacity. Potassium has a significant impact on flower. The histonic contains good potassium due to addition of banana as a source material. Therefore, the highest number of flowers due to absorbable potassium covered by histonic foliar spray. Finally, recommendation. The application of 1% histonic and 50% inorganic fertilizer for high tomato growth and production compared to the recommended rate. The application of 1% histonic with 50% inorganic fertilizer and reduce inorganic fertilizer usage by 50% which will benefit economically and environmental climate as well. Website reference. Finally, thank you for your attention. Thank you.
Okay. Okay, thank you very much, Miss Nishantini. And now the presentation is open for the discussion.
Yeah, Ms. Nishantini, hope you can hear us. Ms. Nishantini, can you hear us?
Yes, sir. I can hear.
Okay, good. What is your control?
Control without water.
Without water, without fish tonic or inorganic fertilizer?
Without water and fish tonic.
Without water and fish tonic?
Fish tonic. Nothing to apply any liquid or solution.
Okay, just water, no?
Just water, yes.
That's right. Yeah, this is my suggestion. Yeah, you can compare that kind of control. It's okay, but it's better to use that most of the farmers utilizing inorganic fertilizer or liquid form or the foliar application for drenching form. So it's better to utilize that inorganic 100% Department of Agriculture recommendation of inorganic fertilizer control. So it makes some value for your research, because then we can say, you can say that this because of that recommendation, your treatment having show some more results, significantly different results compared to that control. So just water is, we know that it doesn't matter. These are suggestions. So based on your final conclusions, 1% fish, no?
Yes, sir.
Okay, then what will happen that increase in the concentration of amino acid concentration, that the results was adversely reduced.
Yes, sir.
Am I correct?
Yes, sir. Yes, sir.
What could be the reason for this?
I think plants absorb correct amount of nutrition. So increase fistonic ratio, maybe it reduce the growth and yield parameters.
Okay, maybe as one reason, refer more, you could have some more that practical reasons on this regard. So thank you very much sharing your great knowledge with us.
Thank you.
Okay, thank you very much, Ms. Nishantini. In that remarks, we will move to the next presentation, next research findings. The topic of that effect of ferrous for acclimatization of tissue cultured plants in hydroponic system. The presenter is Ms. Rana Singha. We will invite Ms. Rana Singha to present her valuable finding with us.
I am Sevani Ranasinghe. It is an honor to present my research to such a knowledgeable audience. I will be discussing about the effect of ferrous in hydroponic system for the acclimatization of tissue cultured cryptocoryne bendy plants. This plant is a member of family Araceae and the common name is water trumpet. According to 2011 research study, the slow propagation by conventional methods, the tissue culture is the most applicable technique for propagation of this plant. The critical stage of hardening process was practiced through the hydroponic system, and as a nutrient, the Albert solution was used in this research study. According to this table, the ferrous content in Albert solution is low in concentration, which is 0.065%. This condition directly affected this plant's growth and development, which led to conduct this research. Ferrous plays a crucial role in biological processes like chlorophyll synthesis, photosynthesis, respiration, nitrogen fixation, as well as DNA synthesis. Therefore, ferrous EDTA was combined with Albert solution to provide adequate level of ferrous in this research study. The main aim and objectives in this research were to examine the effect of ferrous supply for enhancing the acclimatization process in hydroponic system on growth parameters as well as the chlorophyll content of this tissue cultured cryptocoryne bendy plant. This research was carried out the National Aquatic Resources Research and Development Agency in 2023. These were the materials used in this research. According to the research in 2020, 0.5 gram per liter concentration of Albert solution was used with ferrous EDTA at different concentration. And also, 5 treatments with 1 control were maintained with 3 replicates in each treatment and control. The number of leaves, maximum root and leaf length as well as leaf area were measured as growth parameters. In replicate 1, the pH range was adjusted as 6.5 to 7, whereas in replicates 2 and 3 were adjusted as 5.5 to 6. And also, referring the method in research of 1989, the chlorophyll content was measured. Finally, the SPSS software was used for analyzing the data throughout the research study. The data for growth parameters such as the number of leaves, maximum root length, and leaf length was measured at 2-week intervals. This bar chart shows the result in week 1. And these are the results in week 3. And also these are the results in week 5. This bar graph shows the leaf area for each treatment and control in week 1 and 5. According to the bar graph, the treatment 1 shows the maximum value of leaf area. Moreover, this bar graph shows the chlorophyll A, B, and total chlorophyll in each treatment and control in week 1 and 5. According to the bar graph, the treatment 2 shows the maximum value in chlorophyll A, B, as well as in total chlorophyll. This bar graph indicates the overall results in week 1, 3, and 5. The blue line represents the number of leaves, the red line represents the maximum root length, and the green line represents the maximum leaf length in this bar chart. When comparing the results in this bar graph, there is a significant drop in treatment 4 and 5. At the same time, the results of treatment 1, 2, 3, and control are almost equal. However, treatment 1 and 2 reveal the best result in growth parameters. Anyhow, when comparing numerically, the treatment 1 was proved as best concentration of ferrous in this research study. In conclusion, it is clear that the high concentration and toxicity of ferrous EDTA were the reasons for the significant drop of treatment 4 and 5. According to the studies of this research, I recommend the hydroponic technique as more suitable method for hardening process of this plant. And also, I suggest to conduct this research study as a long-term process and applying for circulating system in hydroponic technique as well. Thank you for your time and attention. I hope you found this presentation informative.
Thank you very much, Mr. Anasingha, for your nice presentation and sharing with us your nice findings. And now the presentation is open for the discussion.
Yeah, Mr. Anasingha, thank you. Two clarification. First one is that you have utilized EDTA, as a base of that chemicals to dilute that ferrous. We know
Yes.
that there are two kind of EDTAs available, sodium and also the calcium. What is the real chemical you have utilized for this? Sodium EDTA or the calcium or magnesium EDTA?
I used ferrous EDTA.
Okay, there are two chemicals now. Ferrous alone. Okay, doesn't matter. Generally, we have to use that sodium EDTA to the absorption mode. Okay, you have to know that. In addition to that, your T1 was that the significant highest result shown treatment. No. Okay,
Yes.
then have you examined the pH range of these treatments? Have you worked with that pH range and also the concentration, or only the concentrations?
No, on all the treatments and control, I adjusted pH ranges, 2 pH ranges. In replicate 1, the pH range is 6.5 to 7, and the replicate 3 and 2, the pH was adjusted 5.5 to 6. As according to the research I referred, they proved slightly acidic to neutral pH prefer for this plant, so that I used these pH ranges. All the pH ranges showed the similar outcomes. So
Got it. This means that you have analyzed that pH range as well, no? Okay, good finding. Thank you so much.
thank you so much, sir.
Thank you very much, Ms. Ranasingha, for sharing with us your valuable findings in the tissue culture field. And now we will move to the final presentation of this session. And that is effect of Moringa oleifera leaf extract on germination and seedling performance of Mavi and Mahamavi varieties in Sri Lanka. The presenter is Miss WHDU Pushpa Kumari. We will invite Miss Pushpa Kumari to present her valuable findings with us.
I am Dinesha Udyanjali Pushpakumari, attached to Faculty of Technology, Eastern University, Sri Lanka. Our research title, Effects of Moringa oleifera leaf extract on germination and seedling performance of Mavi and Mahamavi varieties in Sri Lanka. So in this, we are mainly focusing about the seed timing technique. Seed timing technique is that can be utilized to increase enhance the seed germination performance and seedling performance. So improve seedling vigor that ultimately increase the yield of various crop under adverse environmental conditions. So in this study, we use two traditional rice variety, Mavi and Mahamavi variety. We all know rice is the staple food for over half of the world. Traditional rice also imported. Mavi variety especially, it's reported higher yield and unfavorable climatic conditions, especially in waterlogging areas with higher yield and with morphological characters. But farmers are encountering challenges with high yielding which rice variety, Mavi variety, because due to their seed germination and seedling performance, poor seedling performance when they are transplanting in the field. So that so we are focusing on moringa plant because it's reported to its wealth of micro and micronutrient, antioxidant and growth hormones. Recent studies have reported that moringa oleifera, that leaf extract that can be used as a seed priming agent to improve the germination performance and seedling performance. So when we discussed about the nutritional composition, that moringa leaves represent a rich source of vitamins, amino acids, and some antioxidants. Many studies have been conducted to evaluate the medicinal value of moringa oleifera, moringa plant, but little information on it as a seed priming agent. So it has been a growing interest in utilizing of biological priming agent to enhance the seed germination and early seedling development, especially under unfavorable environmental conditions. The main objective of this study to evaluate the impact of moringa oleifera leaf extract on the germination and seedling survival of selected Mavi rice varieties in Sri Lanka. We used two rice varieties, comes under Mavi and Mahamavi variety collected Rice Research and Training Institute, Batalagode. Then sterilized rice is primed in the range of concentration 10%, 20%, 30% and 40%. Here we test two timing periods, 18 hours and 25 hours. Then evaluated the germination performance, germination percentage, hypocotyl length, radical length, seed vigor index, mean germination time, germination index. After following the germination test, seeds underwent priming for duration of 24 hours because we identified it is the optimal priming period. So hence seeds underwent 24-hour priming process with vary concentration. Then after it transferred into the pots in RCTT design, unprimed seeds served as a control. Data were collected based on the seedling performance, especially shoe height, total fresh weight, and data were analyzed in SPSS statistical software following the 2K test for the mean separation. Now I am going to discuss about the results and discussion. We assess germination performances as well as seedling performances. When we look onto the germination performances, when we discuss about the Mavi variety, it reported significant positive results in terms of germination percentage, hypocotyl length, radical length, seed vigor index, mean germination time and germination index. Effect of moringa leaf extract on seed germination. So application of 30% of moringa leaf extract for 24 hours shows the higher seed germination percentage compared to other treatments. In this graph, you can see that dark blue color shows the 24 hours priming period. and 30% give higher germination percentage. Seed vigor index also highest recorded by 30% at 24 hours priming. This graph explain about the highest seed vigor index. It's statistically significant with compared to the treatments. Then highest germination index also reported by 30%. Highest mean germination time also reported by 30% of extract concentration. This graph about the germination index. Now I am going to discuss about the seedling performances. We evaluated the shoot height and total fresh weight. That both parameters higher positive results were reported under the concentration of 40 at 24 hours priming. And now I am going to discuss about the other variety, Mahamavi variety. There is significant effect of tested moringa leaf extract concentration on the germination performances as well as seedling performance. The seed vigor index, germination index also higher under 40% concentration, which reported the higher value in contrast to the other treatments over the control group. These results indicate that 40% of concentration has a positive impact on both seed germination and early seedling growth of Mahamavi variety also. This is the graph which shows the highest value of germination percentage, seed vigor index, germination index. And now I'm going to compare the two varieties of seedling performance under different concentration. In red color, you can see that Mavi variety reported the highest shoot weight, highest shoot height and total fresh weight under 40%, and Mahamavi variety from 30%. Finally, we can be comes to a conclusion that the study illustrated the significant enhancement in germination performance and early seedling growth of Mavi variety through 24 hours priming. The optimal results including the highest germination percentage, hypofertile length, radical length, seed vigor index, and germination index were achieved at the 30% priming concentration. In Mahamavi variety, that positive results come up with the 40% of priming concentration and 24 hours period. So these results emphasize the efficacy of moringa leaf extract priming in enhancing the seed germination and seedling growth. These are the references that used in this study, and thank you very much.
Okay, thank you very much, Ms. Pushpkumari, for your nice presentation. Now the presentation is open for the discussion. I have a small clarification from you that you selected that Mavi and Mahamavi varieties for germination testing. That means that your research, why you are selecting that? Are there any germination issue for these two varieties?
Yes, madam. The recent literatures as well as with the farmer discussion, we had a field survey for the previous study. Then most of the waterlogging areas that farmers claiming that they are not utilizing these, actually not utilizing, there are lack of, how to say that, interest on these traditional rice variety because of some poor germination behavior. That's why we selected these two varieties.
Okay, thank you.
Yeah, Ms. Pushpkumari, one clarification need from you again. Where you have conducted this research, what is the station?
We got the rice accessions from Pathalagoda Rice Research Station, and we conduct the experiment in Faculty of Technology, Eastern University premises.
Good. It means that the moringa leaf extract could be innovation for the field of agriculture in future as well. So try to make some commercial product through this. You have to examine again and again, make some broad varieties of rice to do this repeat session. So then you have to make a good production. Thank you very much. It's nice. Thank you.
Okay, thank you very much, Ms. Pushpkumari, for your nice findings and sharing with.
Thank you.
Okay, on that remarks, all the presentations are over now, and we are going to conclude the session 1 of crop production and protection technologies. And thank you very much for all the presenters.
Thank you all. First and foremost, I would like to thank to the Dr. Nisansala Vidanapatiran and Mr. Hasanaka Rohanadira to conduct the session truthfully. Their valuable comments will help the researchers to develop in the future. Next, all the presenters who share the research findings with us is greatly appreciated. Therefore, thank you very much. All of you are engaging with the session. So we conclude the session of crop production and protection technologies now. Further, stay tuned with us for the next session. Thank you very much, all of you.
Welcome you all to the technical session 2 of the production and protection technology. Members of the technical session 2, Dr. Chaturdinet Pereira, lecturer, Department of Agriculture Biology, Faculty of Agriculture of Ruhuna. Dr. Pereira holds a degree of Doctor of Philosophy in Science from Wrocław University, Poland, and Master of Philosophy in Agricultural Biology from University of Ruhuna, Sri Lanka. Also, and a bachelor degree of Science of Agriculture, special degree from University of Ruhuna, Sri Lanka. Dr. Chatura has received numerous academic awards, including honor, distinction for their doctoral dissertation, and the 12th edition of the Best PhD Foreign Student Competition. He has also been recognized for his research interest, including the Dean's Award for the Best Overall Third Year Students of the Faculty of Agriculture. Dr. Chatura received scholarship from various programs, including Erasmus Plus Blended Intensive Program in Food Sustainability and Climate Change at the University of Parma, and from program, he has also been awarded a PhD student scholarship, Wrocław University of Environmental Science, Environmental and Life Science, and also received fellowship from the Netherlands Fellowship Program for international courses on integrated pest management and food safety. Our panel member, Mr. Sandun Pieruan, lecturer, Department of Soil Science, Faculty of Agriculture, University of Ruhuna. Mr. Pieruan is dedicated scholar with profound academic background and extensive research experience in soil science. He holds a Master of Philosophy degree from University of Ruhuna, specializing in soil science. Following his earliest attainment of Bachelor of Science of Agriculture from the same institution, his academic journey has marked by a keen interest in various facets of soil science, including soil water dynamics, climate change, impact on soil, heavy metal contamination, soil organic matter dynamics, and mineralogy. Dear sirs, the panel is over to you.
Yeah, thank you, Mr. Sirivardhana, to introduce me for this second session of the crop production and protection technologies in the 5th National Symposium of Agro-Technology and Rural Sciences 2024. And thank you very much for the committee to invite me as chair in this session. And we have the panelist Pierre, also happy to share this session with him. Without wasting the time, I think you have the 5-minute presentation already recorded. After that, we have 3-minute discussion in each presentation. And I also invited to the audience to ask any questions and clarification with the centers after their presentation. I invite for the first presentation from the Mr. N. Sarlan. He is going to present of the influence of nitrogen fertilizer regime on the tea yield. It's a meta-analysis. It's now I would like to invite Mr. N. Sarlan as well.
Hello everyone. Today I'm going to discuss about my research topic, that is influence of nitrogen fertilizer regime on tea yield. It was a meta-analysis. These are my contents. So we know tea is the most usable beverage next to the water worldwide. So it's very much famous because of its flavor and health benefits, and is facing the global demand involved optimized cultivation practices for enhanced yield and sustainability. So for each and every plant, we need a certain amount of the nitrogen. So nitrogen is essential for plant growth, significantly affect tea productivity. But there is a huge challenge to manage the controlled fertilizer boost yield, while imbalance use can reduce quality. So I did a meta-analysis involved the quantitative synthesis of data from the relevant studies with a focus on identifying patterns and the trends in the data. So I hope this research can help to inform the policies and strategies to promote the sustainability cultivation practices to the positive impact on the tea industry, and it will help for the environmental impact. So there is a limited understanding of the influence of nitrogen fertilizer regime and the soil properties on tea yield and the quality hampers optimal practices, and inefficient resource utilization, lower tea yield and quality, and the negative environmental impact also the reason for this research. So to address this gap is very much important to improve the tea cultivation and maximize the economic benefits of the tea. It is definitely helpful to the farmer. To analyze the influence of nitrogen fertilizer regime on tea yield and the nitrogen utilization efficiency is the main objective of my research, and to investigate the influence of the nitrogen level and the fertilizer type on the yield and nitrogen utilization efficiency are the specific objective of my study. So for the methodology, my meta-analysis was conducted literature search, screening, construction of database, data analysis, and the data interpretation. I selected 681 papers from various databases like Science Direct, Scopus, Taylor and Francis likewise, and I used these keywords to search my paper. From the 681 papers, I only selected 32 paper by Prisma method screening. So for my analysis, I remove the outliers from the data set, and the significant difference between calculation I used Kruskal test, and the mean separation done test was performed. All analysis was done by using the RStudio software, and the funnel plot also practiced for the identify the publication bias according to the data. So these are the results I got through my research. It's a yield and fertilizer level. There is no insignificant difference. For the fertilizer level and the yield, there is a significant difference. The nitrogen utilization efficiency versus the fertilizer type, there is no any. In the funnel plot also, I got this funnel plot for identify the publication bias in my research data point. From my data points and analysis, I can conclude my research. There is a combined application of organic and inorganic fertilizer. It can be increase the yield than the organic application alone. And secondly, there is no impact on nitrogen utilization efficiency at different levels of nitrogen fertilizer application and the different types of fertilizer. So by overcoming these conclusions and considering this kind of things, we can make a good tea cultivation in future, and it will be helpful to the farmer to maintain the good nitrogen fertilizer level among the tea fields around the world. Thank you.
Thank you very much for the nice meta-analysis presentation. Do you have any question from the Mr. Saralan from the audience? Please raise your hand.
Mr. Saralan, I have a small question regarding that.
If you have. Oh, yes, clear one.
What are the conclusions or suggestions you can suggest for the Sri Lankan tea growers from your meta?
First of all, good afternoon. Instead of Mr. Saralan, I have joined here to the discussion. I'm Intujan. So, yes, there are so many suggestions from my research. First of all, there is a huge problem in identify the correct timing and the nitrogen levels to apply the tea fields in Sri Lanka. So from my research, I can suggest to the farmers in Sri Lanka, they can use the correct timing and correct fertilizer levels of nitrogen to get the good quality and increment of the yield in tea. Rather than that, by analyzing these soil types and the nitrogen levels, it will be help to the overcome the climate differences. Nowadays, Sri Lanka is facing the huge climate impacts. So by analyzing and by overcoming these nitrogen fertilizer regimes and fertilizer levels, we can overcome these climate issues. Like that, for my research, I am not considering the cultivars different. There are so many cultivars in Sri Lanka, but by using this research results, we can build a future research by the different cultivars like TRA cultivars are there. So we can approach this research to other cultivars. We can differentiate the cultivars and do the analysis, and it will helpful to the Sri Lankan farmers, especially tea farmers.
Okay, for the last question, I would like to ask two questions from you. What is the recommended level of the Sri Lanka using the nitrogen level, and what are the most nitrogen use efficiency in the other countries? And the second question is, you mentioned that many articles shows that organic fertilizer has increased the yield of the tea. And what kind of organic fertilizer they used?
Actually, sir, that is the problem. That is a limitation of my research. I couldn't be able to find that kind of results. That is one of the limitation to my research. So in Sri Lanka, we are using as organic compost. We are using the compost, but unfortunately, I cannot be able to find the exact organic fertilizer and the exact fertilizer level. That's why I choose the 300 above 300 and below 300 level. I choose in the range. So in future, we can find the exact level, and it will be help to the find the exact level of fertilizer, especially nitrogen, to the fields.
Thank you very much for the presentation, Sarlan. Because time is going very fast, so let's move to the second presentation of this session. The presentation is effective weed control method for cyperus rotundus in irrigated land of low country dry zone Sri Lanka. The presenting author is Miss Vesna. Over to you, Vesna.
Good morning, ladies and gentlemen. I am Vesna. Today I am here to present my research project. It is effective weed control method for cyperus rotundus in irrigated land of low country dry zone of Sri Lanka. This is the content of my presentation. Next, I will give a brief introduction of my research. Cyperus rotundus is a highly troublesome and economically damaging weed. Often considered one of the worst weeds globally, it propagates mainly vegetatively through an extensive network of tubers. This weed has been reported to cause 20 to 90 yield losses in various agronomic and horticultural crops across the world. Next, I will the problem justification. Cyperus rotundus is one of the worst weeds in the world. High growth rates and quick emergence of these species create competition during early crop development. Farmers use several methods to control this weed from the field. Therefore, this research aimed to identify an effective management method to control cyperus rotundus. The main objective of my research is to identify an effective control method for cyperus rotundus, and the specific objectives are to identify the effect of tillage on management of cyperus rotundus and to identify best management practice or practices to manage the cyperus rotundus. The experimental location was 2CA ARS farm, and the materials that I used for my research are primary and secondary tillage implements, herbicide, black polythene, paddy straw, and herbaceous cover crop with allelopathic properties. The experimental design was split plot design with 3 replicates. There were 2 factors, main plot factor and subplot factor. There were 3 levels in main plot factor and 5 levels in subplot factor. These are the factors, factor levels. These are the treatment combinations. This is the field layout of my research. Next, experimental procedure. First of all, a land area that contains cyperus rotundus was selected. Then, the selected land was separated into blocks. The blocks were divided into main plots. Then, primary and secondary tillage were done using this screw and rotavator. After that, the main plots were separated into subplots. Each subplot was 2 meter into 1 meter in size. Then, the treatments were applied. Here you can see treatments allocation. Next, data collection and analysis. The cyperus rotundus density was measured in 3 randomly selected places using 20 into 20 centimeter quadrant. Biomass of cyperus rotundus was measured in destructive method. Wheat density was measured weekly and the biomass was measured monthly. Then the data was analyzed using SAS software and the means were compared using Duncan's multiple range test at a 0.05 probability level. Then I move to research and discussion part. Here you can see effect of different tillage and management methods on rate density of cyperus rotundus. There is a significant interaction between treatments. According to the results, during first and second month, the lowest weed count was observed in treatment 2, treatment 3, treatment 7, and treatment 8. It means primary tillage with organic mulch, primary tillage with inorganic mulch, secondary tillage with organic mulch, and secondary tillage with inorganic mulch. And the highest weed count was observed in treatment 9, treatment 10, treatment 14, and treatment 15. Here you can see effect of different agronomic practices on weed density of cyperus rotundus. The first graph shows the effect of tillage. According to the results, there is a significant interaction between different tillage methods. Both first and second month, the highest weed count was observed in primary, secondary tillage and zero tillage, and the lowest weed count was observed in primary tillage. Therefore, the primary tillage is most effective than other tillage methods. And the other graph shows effect of different management methods on weed density of cyperus rotundus. According to the results, there is a significant difference between treatments. The highest weed count was observed in cover crop and control, and the lowest weed count was observed in chemical, organic mulch, and inorganic mulch. So these three treatments are suitable for control of cyperus rotundus. There are no significant difference between these treatments, but comparatively lower weed count was observed in organic mulch than others. This graph shows effect of tillage and management methods on biomass of cyperus rotundus. There are significant interaction between treatments. The highest biomass was observed in treatment 10, treatment 10 and treatment 15, and the lowest biomass was observed in treatment 2 and treatment 3. It means primary tillage with organic mulch and primary tillage with inorganic mulch. Here you can see effect of different agronomic practices on biomass of cyperus rotundus. The first graph shows effect of tillage. There is a significant difference between treatments. The highest biomass was observed in primary with secondary tillage. It was followed by control, and the lowest biomass was observed in primary tillage. Therefore, primary tillage shows best performance on control of cyperus rotundus. The other graph shows effect of different management practices control of biomass of cyperus rotundus. According to the results, there is a significant interaction between treatments. Both first and second month, the highest biomass was observed in cover crop and control, and the lowest biomass was observed in chemical control, organic mulch, and inorganic mulch. These three treatments are suitable for control of cyperus rotundus, but comparatively, the lower amount of biomass was observed in organic mulch and inorganic mulch. It means paddy straw and black polythene. In conclusion, I would like to say tillage method with mulching showed better performance in controlling cyperus rotundus. Primary tillage with organic mulching showed the highest performance in controlling cyperus rotundus. At the same time, primary tillage with glyphosate and primary tillage with inorganic mulch also showed better performance than cover crop. Cover crop showed lowest performance. These are the references. This is the end of my presentation. Thank you for your attention.
Thank you very much for the nice presentation, Ms. Vesana. Presentation is open for the discussion. I think we can have only the one question, maybe from the Sandun. Do you have? Oh, maybe I will ask one question because you compare with the primary tillage and the secondary tillage and primary tillage. In the second treatment, according to my observation, you use the primary tillage and secondary tillage both together. But in the results, you observe that only the primary tillage significant reduction of the weeds. So why? Because we are disturbing 2 times in the soil in the secondary tillage and primary tillage. Do you have any idea why weeds are more higher than in the primary tillage when combined with the primary and secondary tillage?
When only primary tillage, the soil was break and turning. But in the secondary tillage, after turning the soil and leveling the soil, then so the weeds, the particles of the weed are turning upper layer of the soil. So the germination is high. That is the reason when primary and secondary tillage done, weed germination is high.
Okay. Thank you for the nice presentation. And let's move to the third presentation of the session. Influence on yield performance of chili through intercropping with legumes. Presenting author is Jema Thuran.
I am Jay Madhuran and I am going to present our research findings on influences on yield performances of chili through intercropping with few grain legumes. The content of my presentation. Before going to the research problem, I will give small introduction about my research topic. Chili is one of the most important condiment crops and widely cultivated throughout the tropical and subtropical countries in the world. It is a major cash crop in Sri Lanka and it has become an essential ingredient in Sri Lankan meals and also has medicinal value too. Capsicum annum is a tropical plant which belongs family to Solanaceae. In Sri Lanka, chili is one of the popular vegetables in most of the dishes. Green chili production is made under 2 seasons, Jala and Maha seasons. In Sri Lanka, the average extent of chili cultivation about 11,000 hectare and production 62 metric ton of green chili. Legumes are nutritious staple of diets around the world. They are an inexpensive source of protein, vitamin, complex carbohydrates, and fiber. Legumes have a range of characteristics that make them a relatively sustainable crop. They can also make their own nitrogen from the atmosphere, thus reducing the application of nitrogen fertilizer. Intercropping means 2 or more crops are cultivating in per unit area and unit time. It's called as intercropping. Therefore, recently intercropping has gained a momentum, particularly in the densely populated countries which has limited per capita land for crop production. Intercropping increases total productivity through efficient utilization of land, labor and drought resources, such as increasing utilization of solar radiation and different inputs, including fertilizer and water. Justification of research problem. Nowadays, you know, the both green and dry chili prices are high level in the local market. Inorganic fertilizer price is also gradually increasing and availability of fertilizer in the market is in poor situation. So we can use maximum utility of land intensity and we can control the weed, we can control the pest and nitrogen percentage in the land. Based on that, this study is mainly aimed to reduce the nitrogen fertilizer requirement and increase the land use efficiency through the intercropping with legumes. Objective, I divided into two objectives, major objective and specific objective. Under the major objective, to identify the suitable legume crops, intercropping with chili cultivation. Specific objective, to find out the effect of legumes on growth and yield of chili. Materials and method. 5 treatments were laid in a randomized complete block design with 3 duplicates and each duplicate consisted of 28 plants. MICH hybrid 1 chili variety was used for this experiment and 3 weeks aged healthy seedlings were transplanted in prepared kilns and each plot 1 seedlings were planted in each kiln and they were irrigated daily and manual weeding was practiced. And chili seedlings spacing 60 into 60 centimeters and plot having 4 into 4 meter dimensions. This is the site location of my research. This is a Tirunelveli research station in Jaffna. Here you can see the layout of the field experiment. This is a randomized complete block design, 3 blocks with 5 treatments. Treatments, T1 was chili, T2 was chili with black gram, T3 was chili with green gram, T4 was chili with cowpea, T5 was chili with DOA recommended chemical fertilizer. Here you can see some figures of my research period. Data collection. I divide it into 2 parameters, growth parameters and yield parameters. Under growth parameters, plant height, canopy width and number of branches per plant. Yield parameters, yield measured by kilogram. Data analysis. Collected data were analyzed using ANOVA procedures by SAS statistical software package version 9.1 and difference between the treatments were compared using Duncan multiple range test at 5% significant level. This is the very important part of our research. So results and discussion. Growth parameters. Under the growth parameters plant height, the results given in the chart describes the intercropping effect of legumes on plant weight. T2, T3, T4 and T5 were significantly increased the plant height during the growing period compared to the control treatment. There was significant difference between the treatments during the experiment period. The significantly highest mean plant height was observed in T5 with compared to the control. There were significant differences among the treatments on mean canopy width through the experiment. According to the treatments, significantly high mean canopy width was recorded in T2, T3, T5 and T4 compared to the control. There were significantly differences among the treatments on mean branches of chili through the experiment and the T4, T5, T2 and T3 treatments were significantly increased mean branches of chili in compared to the control. There is a significant difference between the treatments on yield at 5% significant level according to the Duncan multiple range test. Treatments 4 and 5 are giving higher yield than treatment 1. Conclusion, cowpea is the most suitable legume crop intercropping with chili cultivation with the treatments tested. Comparatively, legumes showed better results on yield performances of chili through intercropping with legumes. This is the references of my research. Thank you.
Thank you for the nice presentation, Jay Mathuran. Now the presentation is open for the discussion. Mr. Firuvan.
Yes, Mr. Jayamathuran, thank you very much for your nice presentation and nice findings. This is not actually a question, just to comment. Are there any plans to promote this intercropping within your farmers, at least in your area, Jaffna? I think you are representing Agricultural Research Institute, no? So as for the results, you got nice results and higher yields. So now, are there any plans to have these results with your farmers, share with farmers? Do you have any plans to share these findings with your farmers in your area, in Jaffna area, as a research institute, agriculture?
Okay, thank you, Sadhumati. One more thing, did you analyze the cowpea or that legume yield with intercropping with the chili? Did you see any difference? So they will show the same amount of yield, the legumes?
Yeah, same yield. No any differences.
Okay, thank you very much for your presentation. Let's move to the next presentation. It's the effect of selected herbal and microbial extraction on severity of chrysanthemum white rust disease. The presenting author is WCMS Madhushankar.
I am WCMS Madhushankar. I am presenting the research on the effect of application of herbal and microbial extract on the severity of chrysanthemum rust disease. Chrysanthemum white rust is a severe disease caused by a fungus called Yersinia horiana. Even though chrysanthemum gives a better yield under greenhouse condition, the high arid condition in greenhouse is favorable for this chrysanthemum white rust disease. But infected plants we cannot sell. Also, growers use chemical fungicides to control this disease, but plants with chemical fungicide residues also cannot export. So, it is an obligatory requirement to find non-chemical method for this problem. The objective of this study to find non-chemical method for controlling the chrysanthemum white rust disease. CRD design was used. Treatment: T1 is control, T2 besides chemical fungicide mancozeb, then T3 turmeric extract and T4 mustard leaves extract, T5 kalwa rhizome extract, T6 fermented fruit juice, and T7 needs solution of fungus and bacteria used in yogurts. Here the look of plants after establishment. Prepare treatment or applied once per every week, and all the treatments were separated using polythene to prevent the mixing of solutions. The permanent fruit juice was prepared using ripened banana and it's mixed with sugar and water 1:1:1 ratio, then kept for 14 days. After 14 days, the solution was extracted and stored. The solution was applied for plants after diluting 5 times. When preparing turmeric and kalwa extract, 100 grams of salt grounded mixed with 1 liter of water and solution applied for plants. Then cultured in 1 liter of water used for washed rice. Mancozeb is a chemical fungicide and applied as the recommendation. In mustard leaves, 50 grams of mustard leaves chopped and mixed with 1 liter of water and filtrate was applied. Disease severity was measured using grid method. 0 to 100 scale was used. 0 is no disease, only 1 covered area less than 1%, 2 between 1 and 10%, 3 between 11 to 25%, 4 between 26 to 50%, and 5 means more than 51% area covered with the diseases. Result and this here the effect of treatment on the chrysanthemum white rust disease at the harvesting. Lowest disease severity reported on The plant used fermented fruit juice and the mango sip and highest cost in the control. According to the mean separation result, there is no significant difference between the treatment, but all the treatment reduced the disease severity significantly compared to the control. Other data of other quality parameters were collected to see the effect, the effect of those how those treatment affect on the quality of the plants. This is how the treatment affect on the plant height. The plants which applied permanent food juice significantly has reduced its height. This is how the treatments affect on the flower diameter. The plants which applied treatment 4, 6 and 7 has reduced the flower diameter significantly. Conclusion. All the herbal extract and microbial preparation had antifungal properties against the chrysanthemum white rust disease as much as mancozeb do, but fermented food juice suppress the plant growth. This is maybe due to high concentration and need to test the best concentration to control the disease. Alpinianigra has antifungal properties to control the chrysanthemum white rust disease without affecting the plant growth, but it is very rare to find the plant. Here are the references, and thank you.
Thank you very much, Mr. Madhushankar, for your nice presentation, and presentation open for the discussion. Sandu, do you have any questions? Okay, I have a small question from Madhushankar. I'm really interested with the yogurt and food culture. What is the that with fermented food extract? What does it final when extraction? Did you analyze the final content of the food extraction, fermented food extraction, or what was it? Because normally, for my understanding, the yogurt and other thing they have some kind of protein sources, so it will increase the the fungus growth. So, do you realize anything?
Not, I didn't, but according to the literature, I found that they are having the antifungal properties. For some studies, both fermented juice and that yogurt extract has been used as antifungal. Has been used its antifungal properties for some studied. Mainly fermented food is having alcohol, but also there are some bacteria and maybe fungus which can use as that we can use that property for it is antifungal property, and also in the yogurt also there are some probiotics and some bacteria and after going in that rice source, this solution also is having some antifungal properties. So I used this to test for the that chrysanthemum white rust also, so to see the effect.
Thank you for the nice answer. And thank you very much, Madhushankar, again for your nice presentation. Let's move to the next presentation. It's on assessing the potential of biochar for increasing water holding capacity of sugarcane growing soil in Sri Lanka. The presenting author is Miss WPD Tilakaratne.
I am Bhumi. The research topic is assessing potential of sugarcane bagasse biochar for increasing water holding capacity of sugarcane growing soil in Sri Lanka. The research was conducted at Agro-Technology Resource Management Division, Sugarcane Research Institute, Udawalawa. Let's see the contents of the presentation. In Sri Lanka, sugarcane are mainly grown in Monoragala, Ampara and Trincomalee districts. Sevanagala, Palwatta, Athimale and Hingurana and also the Kantale areas are cultivate the sugarcane. Sugarcane cultivation practices are mainly two types. Those are rain-fed and irrigated methods. Palwatta and Athimale used to totally rain-fed methods for the sugarcane cultivation, and the Sevanagala area use approximately half amount of rain-fed and other half amount used for the irrigated methods for sugarcane cultivation. Hinguran area use totally irrigated methods for the sugarcane cultivation, and in Sevanagala, recommended irrigation interval is 9 days, and the Hinguran area recommended irrigation interval is 6 to 10 days. So annual total irrigation cost can reduce through increasing the irrigation intervals as directly reduce the number of cycles. So one strategy for this matter, we can increase the water holding capacity of soil. Let's see what are the problem statements of this research. So through this research, we explore the is it possible to improve water holding capacity of sugarcane growing soil in Sri Lanka using biochar, and also is it possible to reduce evaporation of soil moisture using biochar, and also the third one, is it possible to increase the irrigation intervals with application biochar. Let's see what are the objectives of my research. The first objective is to evaluate the effect of biochar application on the water holding capacity of reddish brown earth, alluvial and non-calcic brown soil in Sri Lanka. The second objective is to explore the effect of applying of biochar on changes in soil moisture evaporation. Let's see what are the materials and methods. When consider about the soil sample collection, Galway plantation was selected for the investigation, and as their soil property variation is very high, so therefore soil samples are collected by stratified random sampling technique, and 60 samples were collected from sugarcane farmers field in Hingurana, comprising 20 samples for each soil type. So when consider about the biochar preparation, sugarcane bagases used for the produce biochar as per the procedure followed by the SR, right? Consider about the experiment design. The experiment. The factor one is 3 soil types, as I mentioned, reddish brown earth soil, alluvial soil, and also the NCB soil. The factor 2 is biochar rates according to the weight basis. There is 8 treatments. It goes to control level 0.5%, 1%, 1.5, 2, 3, 4, and 5 percentages. And also use 3 replications. Let's see process of 5 was followed. Spatial distribution of RBE, alluvial and NCB soils plotted on the group based on the research finding of SRI research officer, and all the samples location recorded using GPS. When consider about the collection and preparation of soil samples, grid layout technique used for the collect the soil samples, and the soil samples are collecting using soil core sampler. The depth is 15 centimeter to 25 centimeter because sugarcane land area plug-in depth is that high. And also then after the samples are packed in polythene bags, labeled and transferred to the laboratory. Then after the undisturbed soil samples are oven dried at 105 Celsius at 24 hours. Then after the crushed and sieved by 2 mm siever. Let's see how to prepare the biochar. 15.87% moisture content with sugarcane bagases used for produced biochar, and also the clean fertilizer was used for the prepare biochar. And then after the bagases spiralized under limited oxygen condition around 45 minutes. Let's see how to determine the water holding capacity of soil. Keene-Rakowski-Cox method was used for the determine water holding capacity, and also the 100 grams of soil used for the each experiment, and the biochar particle size is approximately 3 millimeter to 5 millimeters was used for the experiment, and then after the biochar mixed with soil samples control level to 5% respectively. The data was analyzed by Minitab software and ANOVA and mean separation performed by the Tukey method, and also use Microsoft Excel for the get graphs and other calculations. Let's see the results and discussion part. When comparisons of water holding capacity of three soil types, there is a significant difference between these three soil types. The highest water holding capacity received reddish brown earth soil. When comparison of biochar rates, there is a significant difference between biochar rates. The highest water holding capacity received 5% of biochar added soil samples. When considered about the interaction of soil and biochar, there is no significant difference between the soil and biochar. The highest water holding capacity received when applying the 5% of biochar for the RBE soil, and it was followed by the alluvial and NCD soils. So one of the main objective of my research is effect of applying biochar on changes in soil moisture. The introduction part I mentioned, Hinguran area recommended irrigation interval is 6 to 10 days, and also use total irrigated method. So therefore we decided to determine the permanent wilting point. So after the determine water holding capacity of final experiment, that means 5% of biochar added soil samples, then after the determine water holding capacity, then those samples are air dried until get conveyed. So as the permanent wilting point, 8% to 11% of moisture content occurred. Here the graphs you can see the moisture y-axis goes to the moisture content and the x-axis goes to the days of the air dried soil. So the 8% to 11% mean the days are increased 16 to 17 days than the control level water holding capacity. So according to these results, can reduce soil moisture evaporation after the applying biochar. So let's see the conclusion part. So each three soil types significantly improve water holding capacity after applying biochar. The maximum water holding capacity RBE soil tested out of the tested these three soil types, and also the comparing the control level with biochar added soil samples. NCB soil show the most sustainable improvement of in the water holding capacity, and then after the applying 5% of biochar for the soil moisture evaporation decrease. It's directly affect for the irrigation intervals. So and also as the current results obtained based on the laboratory level studies, so therefore we need the field trials as make recommendations for the farm level applications, and also need the further studies for the mass production of sugarcane bagases biochar, aiming at the reducing cost of production before introducing these technologies for the farmers or the sugar industry. So these are the references, and thank you all of you.
Thank you, Ms. Tilakaran, for the nice presentation. The presentation is now open for the discussion. Mr. Sandar. Sandar.
Yes, Ms. Tilagaratna, thank you very much for your nice presentation and nice findings. Do you have any idea about what is the organic matter percentage in your district soils in the Gudan area?
Sorry?
Organic matter percentage?
Actually, it around 1% to 2%.
1 to 2%.
Yeah.
And facilities to increase the production of biogas for the future needs.
Sorry, sir. Sorry.
Do you have facilities to produce in large scale?
Yes. Yeah. In that area, they cultivate mass sugarcane production.
Yes, I am asking about the facilities, laboratory.
Yeah. Yeah, they have. In Sugarcane Research Institute, they have facilities.
Then you can do the further research with the farm level. No? Right.
Okay, I have one.
Okay.
Do you have any questions, Mr. Sanders?
No.
Okay, I have one question. When you are making some treatment, you need to make some consistency. I saw you are starting 0.5, 1, 1.5, after 2, 3, and 5. And I also recommended, I suggest to do them more because it's your maximum point is 5. You are getting the highest observation in the when 5% level. I think it's better to you can the next concentrations also. And actually, I had the same question with the before. It's better to analyze the organic matter content in the each soil sample because it is also affect to the water holding capacity. And for the final graph also, it's better to make some kind of repeated measurement analysis to compare with the water reduction in the tree, and we can take some kind of good point to evaluate which percentage for each soil, which percentage is more important because the different soils are giving the difference actions. So I think it's better to analyze differently for the different soils. And thank you very much for the nice session. Let's move to the next presentation. Author is Devani.
Good morning all, I am Navada Devani, here to present research finding on development of nitrogen fertilizer recommendation for hybrid tomato. Now let's see introduction of the study. Tomato is the one of the most important vegetable plant in the world. Hybrid tomato varieties are often bred for improved yield, disease resistance or other desirable traits which can affect their nutrient uptake and fertilizer needs. Now let's see problem justification of the study. Hybrid tomato varieties require different nutrient amount compared to open pollinated varieties due to their genetic makeup. Currently used fertilizer recommendation is test only for open pollinated tomato plants. There is a lack of research focused on required fertilizer for hybrid tomatoes. This study aims to develop a nitrogen fertilizer recommendation for hybrid tomato varieties grown in Sri Lanka. Now let's see objective of the study. General objective to develop nitrogen fertilizer recommendation for hybrid tomato. Specific objective to study the growth parameters of hybrid tomato under different nitrogen fertilizer level and to study the impact of the application of nitrogen fertilizer on yield of tomato. Now let's see materials and methods of the study. Experiment was conducted in the greenhouse at the Horticulture Crop Research and Development Institute, Gannoruwa, Peradeniya. Factorial complete randomized design was used. Now let's see treatment combination of the study. All these treatments were applied 4 times to hybrid tomato varieties Bhatia and Maheshi. Now let's see nursery preparation. After pot preparation and plant establishment. Now let's see application of treatments of the study. Basal applied urea, TSP and MOP 2 weeks after planting, 4 weeks after planting, 6 weeks after planting, 8 weeks after planting applied urea and MOP. In the data collection, following data were collected. After data gathering, data was analysis using SAS statistical software. Mean separation was done using Duncan multiple range test. Now let's see result and discussion. There was no any significant interaction effect of fertilizer and variety on plant growth parameters yield. Now let's see the plant height at the 3 months after planting. There was a significantly effect of fertilizer on plant height. Significantly higher plant height recorded from F3 and F4 compared to the other treatment. Number of flowers per plant. The significantly higher number of flowers were recorded from all treatments compared to the zero fertilizer. Number of fruits per plant. There was a significant effect of fertilizer on number of fruits per plant. The significantly higher number of fruits were recorded from F4. Root length. There was a significant effect of fertilizer on root length. Significantly higher root length were recorded from F4 and F6 compared to the other fertilizer. Root weight. There was a significantly effect of fertilizer and variety on root weight. Significantly higher root weight recorded from F4 and F5 compared to other fertilizer. Significantly higher root weight were recorded from Maheshi variety. Shoot weight. There was a significantly effect of fertilizer and variety on shoot weight. Significantly higher shoot weight were recorded from F4 and F6 compared to the other fertilizer. Significantly higher shoot weight recorded from Bhatia variety. Yield. There was a significantly effect of fertilizer on yield. Significantly higher yield recorded from F3, F4, F5 and F6 compared to the other treatment. Now let's see result of the study. There was not any significant interaction effect of variety and fertilizer on growth and yield of tomato plants. The significantly highest number of fruits and shoot weight was recorded from F4. Compared to F1 and F2, the yield was significantly higher in F4. It can be concluded that the 90 kg per hectare urea and Department of Agriculture recommended TSP and MOP fertilizer will be verified for development of nitrogen fertilizer recommended for hybrid tomato. These are the reference. Thank you all.
Thank you very much for the nice presentation, Ms. Devani. And this presentation is open for the discussion. I have one question. What is the recommendation dose from DOA for the tomato for the urea?
Basal 65%, 65 kilogram per hectare.
Yeah, why did you use 60? In your treatments, you use the range, you didn't use the 65, but you use 60, 90, 120. Why didn't you go with the 65?
Actually, higher yield and higher growth parameters performance 90 gram per hectare urea 60 and 90 range range include 60 kilogram per
Okay, because they have you already mentioned in your presentation you use the Department of Agriculture recommendation, but in your treatment you use the different concentration level to compare with the other nitrogen levels. That's why I'm asking. And did you use the same percentages for first crop dressing during the vegetation of the tomato plant, hybrid plant? Because normally we are using 3 times urea for the tomato plant, for the basal dressing, first and second crop dressing. Do you use, do you do this, did you do the same thing during your research?
Same
Okay.
apply.
Okay. Please verify with your statistics, because you are talking about the interaction in your results, but you did the one-way ANOVA. So normally for the interaction, you need to do the two-way ANOVA because you are working with the 2 factors. So please consult the statistician before analyzing next time. Okay. Thank you very much, Divani. We can move for the next presentation. Is the final presentation of this session, effect of gamma radiation on seed germination of Catranthus roseus, Madagascar periwinkle. The presenting author is Mrs. S.A.P. Nelka. Over to you.
Greetings to everyone. I am Nelka Srivarnasithya and I am going to share my research findings under the topic of effects of gamma irradiation on seed of Catranthus roseus, Madagascar periwinkle. Let's move to the introduction. Catranthus roseus is an flowering ornamental plant, earlier known as Vincarosia. When we consider about the crop improvement, mutation induction is one of the ways to create varieties by changing a single or more character without affecting the overall phenotype. Physical mutagenesis is an effective mutational breeding method for improving various morphological characteristics of ornamental plants. The most energetic form of electromagnetic radiation is gamma rays and considered as the most penetrating rays when compared to other radiations. Problem justification. Catranthus roseus is currently used as an ornamental plant and still it holds a significant potential for further development in this role. Therefore, this study was designed to evaluate the effectiveness of gamma irradiation as an initial step in applying physical mutagens to Catranthus roseus seeds. Objectives. This study was designed under the main objective is to investigate the effect of various dosages of gamma irradiation on seed germination of Catranthus roseus. Specific objective was to study the germination pattern of gamma irradiated seeds with the time. This experiment was conducted in CIR's research field under the complete randomized design with the 6 treatments and 4 replicates. 6 different gamma ray dosages were used as the treatments. 0 dosage were considered as the control, which was seeds are not exposed to gamma irradiation. Other treatments were T1 to T5 from gamma ray dosages 100 gray to 500 gray respectively. Methodology. Matured seeds were collected from the healthy mother plants and seeds were dried under the sunlight and stored in a clean glass containers. Then seed samples were prepared into 2x2 transparent polythene bags and labeled with each gamma dosage. Then the seeds were exposed to gamma rays using cobalt 60 gamma irradiation source at Horticultural Crops Research and Development Institute, Gandarwar. Then the seeds were transferred into the lab and treated with fungicides and planted them under the shade house condition. Every management practices were done and data collection was done with the daily observation. Data were analyzed using meta-observing statistical software with one-way ANOVA and 2P tests were performed to identify significant differences among the treatments. These are some pictures of the experiment. Let's move to the results and discussion. Results discussion. This figure will show you the statistical analysis of effect of gamma irradiation on seed germination with the time, and the results revealed a significant difference in germination rates among the treatments with the time. The germination rate of the seeds demonstrated a progressively increasing trend over the time across all treatments and reached a maximum value after a constant germination percentage at day 10. All the treatments were started to germinate at day 3 except T5. T5 was shown a delay germination initiation after 5 days from the planting, and it was started to germinate at day 6. And treatment 4 and 5 showed a significantly lower germination percentage in every day when compared to the control and other treatments. 400 and 500 grade treatments resulted in significant reduction in germination, whereas lower dosage less than 400 grade did not exhibit such effects. The highest germination rate was observed in control seeds as 84%, and the lowest germination rate was observed in the treatment 5, which is treated with the 500 grade, as at 8%. Observations are same as I explained in the previous slide. When we consider about the previous studies, the observed results are consistent with previous studies that have demonstrated the negative impact of high doses of gamma radiation germination. However, that gamma radiation at low doses can enhance seed germination by inducing beneficial mutations or activating certain enzymes involved in germination process. At the higher doses such as 400 Gray and 500 Gray, in this experiment treatments, radiation can cause significant damages to the DNA and cellular membranes, leading to delayed germination initiation and reduce overall germination percentages. Similar research findings were shown when increasing gamma radiation doses led to a significant decline in the germination rates of various plant species, and the gamma irradiation induces oxidative stress into cellular damage and reduce germination efficiency. Conclusions. The research indicates that while low dosages of gamma irradiation might not severely affect seed germination, and higher doses such as 400 Gray and above are detrimental to Tetranthes process seeds, these findings have practical implications for the use of gamma irradiation in plant breeding and seed preservation, emphasizing the need to carefully optimize radiation doses to avoid adverse effects on seed germination. These are the references. And thank you, everyone.
Thank you very much, Nilna, for the nice presentation. The presentation opens for the discussion. Okay, I have one thing to clarify. How many hours do you expose to the gamma rays? We cannot hear you, Nilka. Please unmute.
Sorry.
Yeah. Okay.
Sorry, sir. That is depend on the each gamma dosage. That means it is less gamma dosage like 17. It's exposed to around 7 seconds. Likewise, it is increasing with the dosage. So I use 100 to 500 Gray. So that is increasing. That is more than the around 500 Gray. It will take 7 minutes like that.
Yeah, I mean that how many minutes or seconds you expose the seeds to the each gamma ray. You mentioned that to come into the each point of the ray, 100 or something, 7 seconds for the 100. I mean, my question is how many times you expose the seeds to the each radiation?
Yeah, that is actually that with the cobalt gamma irradiation unit, it is adjusted that we have to adjust the rate, that gamma dose. Then it will automatically get the time period with the machine.
Okay, no. I mean, the seed export, when think about 100 radiation, with the
Yeah.
100 radiation, how long you expose the seeds to the 100 radiation? 2 seconds or 5 minutes or 1 minute?
It is around 1 minute, sir.
Okay, okay. And what is the purpose of doing the gamma radiation for this ornamental plant? What is your purpose?
Actually, that Catharanthus process is still have high demand as a ornamental plant. So that if we can improve its morphological characteristics further, so we can introduce as a domesticated plant to the as ornamental, and also that Catharanthus process has some high resistance to in drought condition. So we can introduce that plant as in drought area as a very good flowering ornamental plant.
Okay. Well, my question is, what is your purpose? What kind of things you are going to improve through the gamma ray irradiation,
That means
the
we
mutations?
can yes.
What kind of
We
mutation
can
do you
improve,
accepting?
we can improve its petal color and number of petals. It has only one petal whorl, so we can improve the number of petal whorls and number of petals or any variation in the petals, and also the the dwarf plants. If we can produce some dwarf plants or and more number of flowers in the plants, those are the expected characteristics actually.
Okay, thank you very much, Nelna. There's time to wind up this session. Very fruitful discussions and fruitful presentations. And when we wind up the first, from the first, we discuss about good meta-analysis to how the concentration of nitrogen utilization, organic and inorganic. And I think it needs to improve what kind of percentage of nitrogen is effectively can be used in Sri Lanka. And for the second presentation, control of the kalanduru, very important for controlling weeds because it's directly we can see of the yield by weeds, but for the area to intercropping with chili with some kind of legume. And for also here we discuss about the different microbial, the organic fungicides to control the chrysanthemum white rust disease. And also for the fifth presentation, he shows of the biochar. It's really good because in the dry zone we are suffering from the water. Improvement of using the biochar, we can improve the water capacity in those areas for the sugarcane cultivation. And for the sixth nitrogen fertilizer recommended hybrid tomato, I think it's good for the future recommendation for the agriculture department. And for the final presentation, he is researching on the gamma radiation for giving some kind of mutation for the Catharanthus roseus species. So we have the fruitful and nice section of the crop production and protection technologies. And I would like to thank you for the panel member, Mr. Chachi. And also thank you for the session coordinator, Mr. AJ MC Rivardhana, for having this session very successfully. And finally, I would like to thank again for the fifth organizers of the fifth National Symposium Agro-Technology and Rural Science. Thank you very much. Over to you.
Thank you very much, sir. I take this opportunity to express my sincere gratitude to our eminent panel members, Dr. PCD Pereira, Senior Lecturer, Department of Agricultural Biology, Faculty of Agriculture, University of Ruhuna, sir, and Mr. Sandun Pieruan, Lecturer, Department of Soil Science, Faculty of Agriculture, University of Ruhuna, for sharing your expertise knowledge with NSATRS 2024. And your presence is great honors to us. And my dear authors, thank you very much for your sharing your valuable research with NSATRS 2024, and good luck for your future endeavors. Now let's move towards the third session of third technical session on crop production and protection technologies. Stay with us. Thank you. Come back to the technical session. Welcome back to the technical session on production and protection technologies. It is great honor to introduce our eminent panel members. This panel will be chaired by Ms. Supipi Navaratna, is a senior lecturer, Department and Head of the Department of Food Technology at University of Colombo Institute for Agro-Technology and Rural Sciences. She graduated from the University of Ruhuna under the specialized area in crop science and completed MPhil in horticulture from University of Ruhuna. She has experience in academia in the field of horticulture and organic farming for more than 11 years. And then I will introduce our panel member, Miss KG Katapayarachi. Miss KG Katapayarachi is a lecturer, Department of Agro-Technology at University of Ruhuna, University of Colombo Institute for Agro-Technology and Rural Sciences. Miss Kasuni achieved her bachelor degree in agriculture from the University of Ruhuna with flying colors, while currently reading in Master of Philosophy in Plant Science at University of Colombo. The expertise is up to you. Thank you very much.
Good afternoon, all of you. Thank you very much, Mr. Chatra Sirivardhana. Now we are going to start the crop production and protection technology session 3. So this session we are going to discuss 8 valuable findings. So first, I would like to invite for the first presentation, effect of different levels of inorganic fertilizer combined with organic protein media on vegetative performance of chili, brinjal, and capsicum. So the presenting author is the Dr. P.U.S. Piris. So I kindly invite to the Dr. Piris to share their valuable findings.
Hello, everyone. So when we apply fertilizer application with the department recommendation rates, we hope to get a better yield. So what farmers think, if they reduce the rate of inorganic fertilizer application, they will get a poor yield. But if they apply excessive rates or increased rates of inorganic fertilizer, they will get higher yield or larger fruits or better performance. Is this going to be true? We are to check this out. So if we are to reduce the rate of inorganic fertilizer for this crop, anyway, we have to compensate the remaining amount of nutrients for this particular crop. So if we add a rich rate of organic amendments for this crop, can we actually achieve the same results? So we are going to check this out in this study. So the topic is effect of different levels of inorganic fertilizer combined with organic potting media on vegetative performances of chili, brinjal, and capsicum. So this study was conducted at University of Sri Lanka, and there were 4 treatments, namely T0 was the control, no inorganic fertilizer at all. T1 was the Department of Agriculture recommendation. T2 was the double rate of the T1, and T3 was the half rate of T1. So to go with these inorganic fertilizer recommendations, we also had a very good, very rich organic-based potting media containing general-based compost, topsoil, sand, decomposed refuse tea, and pollinator mixed into 1:1:1:2 ratio. So these ones were arranged in complete randomized design in a net house. So these were the improved varieties of all the crops we used, and the inorganic fertilizers were added at the department recommended rate and the department recommended time intervals. Okay, so actually we wanted to check vegetative performances and also the reproductive parameters and yield of this trial. But unfortunately, after 10 weeks of planting, the plants, some of the replicates died because of the heavy rainfall, so that we couldn't actually analyze data statistically. So we had to conclude the results only for the vegetative performances. So we concluded the measurements 10 weeks after planting. So let's see the brinjal first. Plant height. So at 10 weeks after planting, 8, 9, and 10 weeks after planting, the highest plant height was observed in D0, where we didn't apply any inorganic fertilizer, compared to T1, which is the department recommendation. T3 was the half rate, so that was significantly different to T0 and T1. So what happened to T2 here? Unfortunately, most of the replicates in T2, which is the double rate, died because after toxicity. So that means we should not apply excessive rates of inorganic fertilizers for this delicate brinjal plant, which can cause toxicities and death of plants. When it comes to chili, so the low highest height was recorded again in double rate of inorganic fertilizer application, compared to all other treatments in all 7, 8, 9, and 10 weeks of planting. To capsicum, there were no significant differences between the treatments for the plant height towards the vegetative phase and at the start of the towards the start of the reproductive phase. So when it comes to number of leaves at 10 weeks after planting, brinjal showed the highest number of leaves in control and T3 treatments, the half-rate treatments compared to department recommendation. Chili, control, and T1, which is the department recommendation, had the highest number of leaves compared to the double rate application, and capsicum did not have any significant differences between the treatments. Conclusions. So based on the present results, for better vegetative performances in brinjal, chili, and capsicum, actually we can go with the reduced rates of inorganic fertilizer application, but we have a very rich organic potting media. But anyway, we should not apply any excessive levels of inorganic fertilizer, which resulted death of the plants and poor performances. So we should inform this information to the grower so that we can save some money, we can save environment, and we can also ensure better plant performance. So these are some of the references. I would like to acknowledge these people and institutes. Thank you very much for listening.
Okay, thank you very much, Dr. Piris, for the nice presentation. So now presentation is open for the discussion. I have a few clarification from you. In here, you have protein mixture, one of the ingredient of the protein mixture. What is the special reason? Yeah, what is the special reason using the fall manure?
Yeah, because it is a rich source of nitrogen, not only nitrogen, phosphorus, and potassium. So I thought because we are targeting to reduce the amount of inorganic fertilizer we add, so that we have to actually give some amount of nutrients, that I added poultry manure as a good, like a rich source of nutrients, because compost was not that rich. So I wanted to add a rich source in place. So I think that is a reason for the significant difference, because control plant actually showed a higher performance when we add inorganic plus the organic matter. Actually, we suspect that there have been a toxicity. So the reason for poultry manure is because it is a rich source of nutrients, especially NPK.
Okay, so I have another small clarification. So according to your result, you are focusing on the control and the T1. So there's a hidden message from the treatment, no? So yes. What is your conclusion for the farming community regarding the treatment 2?
Yeah, of course. T2 is a double rate of the department recommendation. So we don't get anything if you apply excessive rates of fertilizer. I mean, we don't get the expected bigger yield, higher number of fruits when you increase the rates of application. Instead, we can just give what is required by the plant, what is demanded by the plant. So we can reduce the amount of inorganic or the chemical fertilizer we add, and then we can save money, environment, and also it will be healthy for us as humans who are going to consume these things. So that will be save of everything. Yeah. So yeah.
That simply over application or usage of the inorganic fertilizer does not give the good result after all.
Yeah. Just utterly waste of money.
Yeah, exactly.
I have small suggestions.
Yeah,
I feel that it is better with this treatment if you can go through the range of fertilizer, not only the 50%, it means half to 75%. Likewise, if you have increased the range, it will be more beneficial.
Yeah, okay, yeah, that's good. So we'll do it in future. Thanks. Thank you for the suggestion.
Okay, thank you very much, Dr. Piris, for the nice presentation.
Thank you.
Next presentation is evaluation of different soil amendment to manage clubroot disease. Okay, so presenting author is Dr. Chitrani Ranasinghe. So I cordially invite to the Dr. Chitrani to present their valuable points.
Good afternoon, everybody. My presentation is on evaluation of different soil amendments to manage clubroot disease in cabbage. First, introduction. Cabbage is the widely grown vegetable in the brassica family in Sri Lanka. It prefers moist, cool weather. However, at present, it is grown every agro-ecological zones of the country. Clubroot disease caused by Plasmodiphora brassicate is one of the major threats in the cabbage production. After infection of this pathogen to the soil, it is very difficult to eradicate because it is a soil-borne pathogen, has very complex cycle. Pathogen survives testing without host run more than 13 years, 17 years. When consider management of this disease, plant-resistant cultivar is the best solution, however not available. Therefore, at present, standard recommendation to manage this issue is raise the soil pH 7.2 or above by adding lime. But there are no recommendation regarding the quantity of lime applied to manage this issue. Apart from the lime, farmers use different materials as soil amendments in different ratios as well as different combinations. However, it was observed not so limited successful. Therefore, this study was conducted to find the effectiveness of farmer practice soil amendment to manage the clubroot disease. Then moving more to the material and method. In this experiment, we tested different soil amendment, different ratios as well as different combinations. For trial and field trial were conducted. Soil amendment were done 2 weeks before planting in both trials. For trial, 12 inches by 12 inches. Side spots are filled with clubroot infected soil as 5 kilograms per pot. Field trials were conducted in already clubroot pathogen infected field. As the collection, pH of the soil were recorded before soil amendment at planting before first, second and third of dressing. Disease severity of the roots were recorded at 3 months after planting using this severity scale. Rainfall data were recorded in the field trial throughout the growing season. Then we will see what are the results and discussion. These two graphs explain the change of soil pH in both trials. According to this, you can see in both trials, except poultry manure amended through treatment, all other treatments showed increasing trend of pH after soil amendment until second crop dressing, and afterward it showed decreasing trend. In the field trial, except lime-added treatment, all showed decreasing trend of pH after soil amendment. Here also, poultry manure amendment treatment showed the lowest disease severity. Compared to the pot trial, field trial recorded low disease severity. It may be during the growing period, it was recorded heavy rainfall. This graph explains the disease severity percentage of both trials. You can see disease severity was recorded similar trend in both trials. Fungicide applied treatment showed significantly lower disease severity. After that, lime-added treatment showed the significantly lower disease severity compared to the other treatments. According to our results, there is no difference in the clubroot disease severity between two trials, even though pH was higher in the pot trial than the field trial. In pot trial, even though pH showed in some treatment, showed no reduction in disease severity. On the other hand, even untreated control also showed similar change in pattern of pH as other treatments. It is clear that pH was not constant throughout the growing season, even for trial conditions. Our results agree with some other previous researchers' findings. Finally, our conclusion of this research is soil amendment with lime at rates of 1.5 per hectare or 2 tons per hectare proved to manage cabbage disease up to some extent. Our suggestion is it is necessary to carry out further research in order to develop effective management strategies for managed clubroot disease. Now, these are some of the references. Thank you for your kind attention.
Okay, thank you very much, Dr. Chitrani, for your nice presentation. So now presentation is open for the discussion. So I have a small clarification from you, Dr. Chitrani. What is the base to select the lime application only the 1.5 tons per hectare and 2 tons per hectare?
Actually, it is the general recommendation, especially in upcountry area, pH of the soil is very low. It is a general recommendation before planting any crop should apply 1.5 or 2 tons per hectare lime, but actually no recommendation regarding the increased pH up to 7.2 in recommendation is general recommendation.
Yeah. So with the application of this rate of the lime, what is the pH value? Is it come to the 7.1 or something?
No. It is not come to the pH level. That's why I checked that amount. pH is come to the 7.2. Actually, we observed that that lime amount is not sufficient.
That means the further study necessary to find out the suitable lime application rate, no? Okay.
Yeah. Okay. According to our observation, amount of lime applied should be increased than that recommendation.
Yes. Okay. Thank you. Right. Okay. Thank you very much, Dr. Chitrani.
Okay. Thank you.
Okay, the next presentation is agronomic method to improve nitrogen use efficiency in rice. The presenting author is Ms. RGD Pratibani Karunathilaka. So I cordially invite to the Ms. Karunathilaka to share their valuable findings with us.
Welcome all of you. I am going to present about my research. My topic is agronomic method to improve nitrogen use efficiency in rice. I am Pratibani Karunathilaka. This is content. Next, introduction. Rice is most important cereal crop in the world, and rice requires significant amount of nitrogen for optimal growth and productivity, synthesis, protein, enzymes, and chlorophyll, as well as nutrient uptake and energy production. When explained problem statement, nitrogen requirement of rice cultivation is given by chemical fertilizer, and nitrogen use efficiency of rice is 30%. About 70% of nitrogen is wasted. Then move to justification. By improving agronomic nitrogen use efficiency, comparatively same yield level can be maintained, low level of nitrogen fertilizer and cost to minimize nitrogen loss as environmental problem, as well as give economic benefit to farmers. The overall objective is to improve nitrogen use efficiency agronomically in rice. The specific objective is to investigate whether agronomic nitrogen use efficiency can be improved by mixing nitrogen inhibitor with urea in rice. Let's move to materials and method. The research was done at CIC Seed Farm Palvera in early season. Now you can see materials. Those are field equipment and laboratory equipment. This table shows treatments. When considered about experimental design, randomized complete box design was selected and there are 9 treatments with 3 replicators. Plant preparation, crop establishment, crop management practices and harvesting were agricultural practices. Let's move to data collection and analysis. Yield component, namely panicles per square meter, spikelets per panicle, field grain percentage, and 1000 grain weight were recorded and measure the dry weight. Then calculate the actual yield and agronomic nitrogen efficiency after the data were analyzed. Let's move to result and discussion. This table shows actual yield and agronomic nitrogen use efficiency. Lowest grain yield shows no nitrogen treatment. According to this table, can see rice responded only up to 75% of recommended dose of urea and 75% recommended dose of urea and 100% recommended dose of urea gave same yield. Grain yield of only applied 50% recommended dose of urea was low, but when applied 50% recommended dose of urea with chemical inhibitor 1 liter per metric tons was increased to 75% to 100% recommended dose of urea in grain yield. When look agronomic nitrogen use efficiency, comparatively higher nitrogen use efficiency saw 50% recommended dose of urea with chemical inhibitor 1 liter per metric tons than 100% recommended dose of urea treatment. Next, looking about each component response for different treatment. Comparatively higher panicles per square meter shows treatment 1, 4, 5 and 7 and low panicles per square meter shows treatment 6, 8 and 9. Treatment 1 shows higher number of spikelets per panicle and when reduced 50% recommended dose of urea show lower number of spikelets per panicle. Not significant 1, 2, 3, 4, 5, 6, 7, 8 and 9. Low field gain percentage show no nitrogen treatment. Treatment 5 shows comparatively high 1000 grain weights and others are not significantly different. This is conclusion. 50% recommended dose of urea with chemical inhibitor 1 liter per metric ton shows the significantly highest agronomic nitrogen efficiency among the treatments. Use of 50% recommended dose of urea with chemical inhibitor 1 liter per metric ton appears to be efficient in increasing agronomic nitrogen efficiency in rice cultivation. Therefore, instead of using 100% recommended dose of urea, using 50% recommended dose of urea with chemical inhibitor 1 liter per metric tons can save 50% recommended urea amount. However, this has to be confirmed by further experimentation and in addition, cost effectiveness of the use of nitrogen inhibitor has to be studied. Those are differences. And thank you.
Okay, thank you very much, Ms. Karunatilaka, for your nice presentation. So the presentation is open for the discussion. I have a few clarifications. What is the difference? You explaining about the nitrogen inhibitor. So then another term is the nitrogen stabilizers. Okay, what is the difference between nitrogen inhibitors and the nitrogen stabilizers? Is any difference?
Is one stabilizer.
Okay, so yes, so in here you use the agronomic method to improve the nitrogen use efficiency now. So can you give any other agronomic practices to improve the nitrogen use efficiency other than the using the nitrogen inhibitors?
We can use clay ball with some nitrogen inhibitor with clay ball, nitrogen inhibitor, other one. Use
okay,
as clay ball.
Okay. So I asking what are the other agronomic practices can be used to improve the nitrogen use efficiency in rice other than using use the nitrogen inhibitors. So there are some more agronomic practices can be used to increase that or improve that. So I ask.
You can use paddy. Nitrogen testing.
Right, okay. Thank you very much, Ms. Karunatilaka, for your nice presentation. Okay, right. Then the next presentation is enhancing germination and growth through seed treatment and media optimization. So the presenting author is Mr. BMKAP Bamunusingha. So I cordially invite to the Mr. Bamunusingha to share their valuable findings with us.
Good day to everyone. I am Colombo, one of the authors for the research article named Enhancing Germination and Early Growth of Passiflora foetida through Seed Treatment and Media Optimization. Passiflora foetida is an important medicinal plant that has been found in tropical regions of the world. It has been widely used for several health issues and having more medicinal value. For cutting, plant cells and tissues could be used to quickly and widely propagate valuable medicinal and commercial plants. This could serve as a backup supply for the large-scale cultivation of common and threatened plant species. This is the research problem. Unfortunately, this fruit plant is not taken for human consumption in Sri Lanka. Therefore, it has become an underutilized fruit. Due to people's ignorance about this fruit, its economic value has decreased. Hence, there is a necessity to propagate adequate amount of plants to increase its popularization. But there are lack of information on successful propagation. While a limited number of in vitro studies, research on seed propagation under field condition is scarce. Hence, considering this, an experiment was conducted with the objective of to establish a rapid propagation technique and identify the most effective germination-inducing agent and medium for seed propagation of Passiflora foetida. The experiment was conducted at the UCIRS Hambantota, belonging to low country dry zone. Seeds were collected from well-ripened fruit in the farm field. This experiment was conducted to identify the best germination-inducing agent with different potting media for seed propagation of Passiflora foetida. The experimental procedure was arranged in a factorial CRD with 9 treatment combination with germination-inducing agent and potting media type. Each treatment was replicated 4 times and each replicate consisted of 10 seeds. These are the treatment combination. All seeds obtained from well-ripened fruit were thoroughly washed with tap water at room temperature and soaked in water for 24 hours and then air-dried and treated. Data action was started as soon as seedlings started to emerge. Percentage days taken for germination, survival percentage, number of leaves from seed were measured. ANOVA was performed for all the data at the 5% probability level using SAS software. DMRT was used for mean separation. Let's move to the result and discussion part of this presentation. The statistical analysis revealed a lack of significant interaction between the tested factor on germination of Passiflora seed. GA3 demonstrated the highest germination percentage, first germination percentage during 7th and 8th days respectively. Furthermore, a significant difference was observed mean type during the 7th and 8th days experiment. Specifically, topsoil and sand with topsoil media displayed the highest germination percentage, while sand media yielded the lowest germination percentage. There is no significant interaction on the days required for 50% germination. However, notable difference emerged in the various seed treatment method during the 7th to 9th days. A significant difference between the seed treatment method was observed with gibberellic acid treatment, demonstrating a lower number of days for 50% germination compared to the hot water treatment and control. There is no significant interaction on number of leaves, but a significant difference was observed among different during the second week. Gibberellic acid seed treatment method exhibited a significantly higher average number of leaves and the lower leaf count observed in the hot water seed treatment method. Furthermore, a significant difference was observed among various media types during the second week with highest average leaf count recorded in the topsoil and sand with topsoil media and lower number was observed in the sand media. A significant interaction was observed on the survival percentage during the second week. Higher survival rate was observed in seed subjected to hot water treatment. Lowest was noted in seed treated with hot water and sand media, as well as in untreated control seed planted in topsoil and a mixture of sand and topsoil media. Let's move to the conclusion. Gibberellic acid treatment notable accelerated germination and increased leaf count compared to the control. Media choice also impacted leaf development with topsoil and sand with topsoil media resulting in the highest leaf growth. Survival rate in the second week varied significantly, with hot water treatment and planting in topsoil showing the highest soil compared to other combinations. These are the preferences. And thank you.
Okay, thank you very much, Mr. Bamunu Singha, for your nice presentation. Presentation is open for the discussion. I have a application. Mr. Bamunu Singha, can we use the vegetative propagation method for the Passiflora foetida? Can you hear us?
Yes,
Okay.
now here.
Okay, right. I repeat the question. Can we use the vegetative propagation method for Passiflora foetida? You use the
Sorry?
You use the sexual exemption, so the
Yeah.
vegetative propagation, any did you find any vegetative propagation method can be used for this plant?
Not clear for your sound, ma'am. Hello?
Can we use the vegetative propagation method for Passiflora potida? Any of the vegetative method?
We can use.
We can use.
Yes, we
Okay.
can use.
Yeah, okay. So then next question. What are the most successful method out of those vegetative method?
It's a most successful method is a cutting propagation method, ma'am.
Okay, cutting. Right. Okay. Thank you.
And I have a small question. So you have mentioned it is a underutilized crop. So can you tell some suggestions to popularize this one? What are the potential uses of this plant to make
Yes, sir.
popularize Sri Lankan?
Yes, ma'am. I conducted with a social survey with this plant. Hello?
So what are the ideas of peoples according to your survey?
Yeah, this plant have a very important medicinal value. It is a an fruit, and it is some people are eating for the vegetables.
Okay, thank you. Thank you, Pamal Singh.
Okay, thank you very much for your nice presentation. Okay. Now we move to the next presentation. Effect of different organic fertilizer application on growth and yield of tomato. The presenting author is Miss Pamina Tangarasa. So I cordially invite to the Miss Tangarasa to present the his presentation.
Good morning to you all. Today I am going to present about my research presentation. My title is Effect of Different Organic Fertilizer Applications on Growth and Yield of Tomato. This is my content. We will move to the introduction part. Fertilizer is a natural or artificial substance containing the chemical element that improve the growth and yield of plant. Fertilizer mainly two types: organic and inorganic. Organic fertilizer is a fertilizer that is derived from organic sources. Organic fertilizer have several advantages. For example, improve the soil fertility, balance the ecosystem, and also improve the soil water holding capacity. Liquid fertilizer refers to any kind of liquid solution that is provided to plant as the form of food. Tomato is the economic important crop. It is a family crop. It's rich in source of vitamin, mineral, fiber, and hydrogen source of antioxidant. Now we can see the research objective to determine the best organic fertilizer that provide the high growth and yield of tomato, to evaluate the growth parameter of tomato with selected organic fertilizer, and to measure the different nutrition composition of different organic fertilizers. Here we can discuss the research material and methodology. In this research used for different treatment with 5 replicates, treatment application of Jivamata organic fertilizer, treatment 2 application of Panchagavya organic fertilizer, treatment 3 application of Amrutagazal organic fertilizer, treatment for control. This is the experimental design for the research. Sometimes complete blood design method. Here we can discuss the preparation of Panchagavya liquid fertilizer. For a certain amount of half urine ghee was put into the butter. The solution was mixed well. The solution was allowed to 3 days for fermentation. After 3 days, a certain amount of half tender coconut water, sugarcane was wrapped in banana was wrapped in the solution, and the solution was mixed well. The solution was mixed thoroughly 2 times by 1 day. The solution was allowed for 7 days. After 7 days, the yeast was added to the solution, and then the solution was allowed to 90 days for fermentation. After 90 days, the solution ready to use. Now we will discuss the preparation of Amadakars liquid fertilizer. First, 1 kilogram of powder, 1 liter of urine, 200 gram of jaggery, and 10 liter of water was put into the bucket. The solution was mixed well. The solution was allowed for 1 day for fermentation. After 1 day, the solution ready to use. Here we can see the preparation of Amadakars liquid fertilizer. First, 1 kilogram of powder, 1 liter of urine, 200 gram of jaggery, 2 gram of gram flour, and 1 handle of soil, and 20 liter of water was put into the bucket. The solution was mixed well. The solution was allowed to 3 days for fermentation. After 3 days, the solution ready to use. Here we can discuss the results and discussion in this chart. So, soil physical chemical properties of the soil. Here we can see the physical chemical properties of the organic fertilizers. Now we can discuss the growth parameter analysis. Here we can see mean height at the week interval. Here T1 show higher significant difference than other 3 treatment. T4 show very low significant difference than other treatment. Now we can see the mean number of leaf at the week interval. Here T1 have a higher number of leaf than other 3 treatment. T4 have very lower number of leaf than other treatment. T2 and T3 have slightly same number of leaf than the 2 treatment. Now we can discuss the mean number of fruits at the week interval. Here T1 show higher number of fruits than other 3 treatment. T4 show a very lower number of fruits than other 3 treatment. Now we can discuss the mean number of branches at a week interval. Here T1 have a higher number of branches than other 3 treatment. T2 and T3 have same number of branches than other 2 treatment. T4 show a very less number of branches than other treatment. Now we can see the mean yield at a week interval. Here T1 have higher amount of yield than other 3 treatment. T4 show very lower amount of yield than other 3 treatment. T2 and T3 have same amount of yield than other 3 treatment. Now we can see the conclusion of the research. It can be concluded that the organic fertilizer is the solution for the tomato because 2 organic fertilizers have maximum plant height, high number of leaf pods, and large amount of yield than other 2 treatments. So the organic fertilizer can be successfully used for increase the growth and yield of tomato. So the organic fertilizer is recommended to the farmer for cultivation of tomato. This is a reference. Thank you.
Okay, thank you very much, Ms. Thangaraja, for your nice presentation. Now presentation is open for the discussion. So I have few clarification regarding your research. Okay, yes, the first one, yes. What is while you are preparing the Jeevamurtam, what is the purpose of application of the soil to the Jeevamurtam?
The fertilizer increases the microorganism activities of the soil.
Ms. Thangarasa, can you hear us?
Yes, miss.
Okay. So, while you are preparing the Jeevamurtam, add the soil, no? Okay. So, sorry.
Foliar application.
No. While you're preparing the Jeevamurtam, not applying as a foliar application. When you prepare it, you add the soil to the Jeevamurtam, no?
Yes, miss.
Okay. So, what is the purpose to apply the soil to the Jeevamurtam? Because of
Jivamrita organic fertilizer increase the microorganism activities, so release the nutrition.
Right. Okay. Okay. So I have a suggestion. Okay, so you only in here you only compare the three organic liquid nutrition solutions. Okay, the amortizal, geomortam, and panchagavya with the control that anything, right? Am I correct? So it is
1:1 ratio of soil and compost first type.
all right. Okay, so it's better to insert the department recommendation as a treatment. You can get the very clear finding what is the yield or the compare with the department recommendation, because of farmers are normally applying the department recommendation. No? So then in your research, you only focusing on the organic applications. Okay. So if you have a possibility to compare inorganic application department recommendation with those organic application, it will much beneficial and give a strong finding for you. Okay, so that is my suggestion. So if any
yeah, I have same suggestion, because to improve the beneficialness of this study, so you need to compare it really with the inorganic fertilizer. If not, that you can't give the idea to farmers, because they are normally using inorganic fertilizer. So before going to the 100% organic, it's better to compare. You can use a positive control, the inorganic fertilizer, it is department recommendation, and you can use the integration of the inorganic and the organic. I think it will be beneficial.
Right, okay. Thank you very much, Ms. Tangarasa. Then we move to the next presentation. Determination of arsenic level in irrigation water and agricultural soil of export-oriented leafy vegetable cultivation. So presenting author is Ms. PHMGS Kim Hani. So I cordially invite Kim Hani to share their valuable finding with us.
Hello everyone, I'm PHMGS Kim Hani, and today I'll present my research on determination of arsenic levels of irrigation water and agricultural soil in export-oriented leafy vegetables Centella asiatica and Alternandra sacchalis. This is the content and facts are lined up in this order. At first, let's move with introduction session. Your plant causes heavy metal contamination of food through root and leaf uptake. Due to this severe environmental pollution caused by the world's economy's increasing industrialization, the majority of agricultural products, particularly leafy greens, have been contaminated. These past 10 years, Gotukola and Mugunuana emerged as popular green vegetables, particularly in European Union's export market. Scope of these toxic heavy metals analysis was the European Union notification of exported leafy vegetables due to the pesticide residues on 26 January 2023. Notification report that the high level of pesticide residues in Gotukola and Mugunuvanna without complying with the relevant requirement. This analysis was conducted to determine toxic heavy metals in agricultural soil samples and irrigation water samples relevant to the export-oriented Gotukola and Mugunuvanna samples as a factor analysis of determination of toxic heavy metals in leafy vegetables to European Union. This is the research objectives. General objective is to determine lead, cadmium, arsenic contents in water samples and soil samples of the leafy vegetable peas, and the specific objectives are to determine farming practice of leafy vegetables and to determine relationship between detected samples, irrigation water and agricultural soil with the previously checked leaf samples. Movie B, materials and methodology. In this study, major raw materials were irrigation water samples and agricultural soil samples relevant to the leafy vegetable samples. Those are the major chemical reagents used. Here I have listed down equipment used and selected metals for the analysis. In here we consider majorly about arsenic because previously arsenic was the only element which has exceeded the maximum permissible levels in Gotukala and Mugunuana. Next, look for the sample preparation. It consists with three major processes: standard preparation, water sample preparation, and soil sample preparation. Here I mention each and every process under standard preparation, soil sample preparation, and water sample preparation. If you are interested, you can go further. Dry method is used for metal analysis of soil samples. So first sample should be allowed for oven drying at Celsius degrees of Celsius for 24 hours and taken for further testing. Under the order to digestion, dilution process, injection, and finally analysis. For the water samples, we use wet method, means we skip the drying part. We can use digested water samples for the further processing as I mentioned. In research and discussion session, arsenic was found in 26.31% of the irrigation water samples, which has higher than the MPLs for the elements. 50% of samples in Putlam district had levels of arsenic exceeding the corresponding MPLs of 0.1 ppm according to the recommendation. The concentration of arsenic in Kalutara district was significantly below according to the WHO or FAO recommendations, and no sample surpassed maximum permissible levels. In here, you can see the detected irrigation water samples in blue color from the total water samples. Based on WHO FAO recommendation, toxic heavy metals MPS were not exceeded in agricultural soil samples from Putlam and Kalutara district. In general, the maximum amounts allowed in agricultural soil are greater than the maximum amounts allowed in vegetable and irrigation water. Here I included a diagram further for your reference as well. In order to examine the effects of irrigation water and agricultural soil on the arsenic content of leafy vegetables, I have included a graphical illustration. Here I insert two-line chart which illustrate about the detection of heavy metals in leafy greens, irrigation water and soil samples and Putlam. As you go smoothly on this line, you'll find there's no any relationship between detected leaf vegetables and water and soil samples further. As sum up, soil of leafy vegetables found in Putlam and Kalutara district do not contain arsenic in concentration exceeding the maximum permissible levels. About 50% of irrigation water source used for leafy vegetables farming in Putlam and Putlam district contain arsenic in higher concentration than the MPS, whereas the irrigation water source used for leafy vegetable farming in Kalutara district do not contain arsenic in higher concentration than the MPS. Certain samples of leafy vegetables intended for export, Alternata sessilis and asiatica, contain arsenic in amount that exceed the maximum permissible levels. The amount of arsenic in irrigation water in Putlam and Kalutara districts has little to do with the amount of arsenic accumulated in leafy vegetables. Here the references according to the citation. This is the end of my abstract presentation. I sincerely appreciate your attention. Thank you very much all.
Okay, thank you very much, Ms. Kimhani, for your nice presentation. Presentation is open for the discussion. So I have a small clarification. So in here, in your discussion, you discuss about the suitable, it should be find out the suitable irrigation water source, especially for the Puttalam district, no? So therefore, because of some irrigated water are contaminated with the arsenic. So then can you suggest the suitable irrigation water source for those district, especially for the Puttalam?
Madam, Puttalam district is a dry zone. They used to apply apply deep well water as irrigation water. They never try to apply tap water because of the cost. So I think it's better to use tap water because already we in it that there is an issue in deep well water with contamination.
Thank you.
I have same question because I need to know what would be the reason to increase the arsenic content in the Puttalam district especially. So you felt that it is due to the deep well they are using.
Yes, ma'am.
Okay, thank you.
Right, okay. Thank you very much for your nice presentation, Ms. Gimhani.
Thank you, ma'am.
Okay, the next presentation is a comprehensive review on RA perspective for pest and disease management. The presenting author is Dr. HBD Kaushalya. So I cordially invite the Dr. Kaushalya to share their valuable finding with us.
Welcome to my research presentation. Today I am going to present about a comprehensive review on Ayurveda perspective of pest and disease management. These are my contents. Ayurveda, an ancient Indian medical system and holistic approach to physical and mental health. Ayurveda, an ancient Indian practice which promotes plant health, soil health, and resilient system through traditional knowledge. Agriculture contributes to economic development, poverty reduction, food security, and sustainable practices like Ayurveda, improving soil health through measures such as natural fertilizer, pest management, and water management. Ayurveda emphasizes community knowledge and participation in agricultural practices and promoting adaptive and resilient systems. Agriculture development is a crucial tool for reducing poverty and contributing to 4% of global GDP. Objective of this study to review Ayurveda perspective on pest and disease management. This study used SLR approach and Prisma criteria to select 64 articles, search terms, pest disease, and Ayurveda control or management. Let's move to the results. Ayurvedic pest and disease management principles emphasize holistic approach, preventive measures, natural remedies, and sustainability. Preventive measures include soil health management, crop rotation, and intercropping, and plant selection. Soil health management is crucial, involving composting, vermiculture, and organic fertilization, which promotes plant growth. Here you can see different types of soil classification according to Ayurveda. In this table, it shows one-off soil classification by considering factors such as dosha dominancy, quality of air, rainfall, nature of land, disease tendency, and suitable plants. This is another classification based on Panchamahabhuta or 5 basic elements. Acharya Charaka states that the soil has a significant impact on the quality of Aushadha and Ahara Dravyas. In this table, it shows the process of planting according to the Acharyas. Ayurveda principles promote crop rotation, combination planting, hand weeding, and herbal weed sprays to maintain soil fertility and reduce weed growth. These are some measures mentioned in Vuksha Ayurveda to control weeds. Kunupachala is a manure made from boar fat, flesh mixed with water and milk. Here you can see different recipes of natural fertilizers. Plant diseases can be classified into two categories and treatment to these diseases done according to the causative factors. In this table, it shows types of endogenous diseases with the features of diseases and treatment. And here you can see some causative factors for exogenous diseases with their treatments. Let's move to the discussion. Vuksha Ayurveda is beneficial for modern agriculture due to it emphasis on natural harmony, biodiversity, and minimal ecosystem disruption. Ayurvedic pest and disease management techniques promote ecological balance, soil health, plant diversity, and cost-effective pest and disease management, reducing reliance on synthetic chemicals. Ayurvedic pest and disease management challenges in standardization, efficacy, and production scaling. So, future research should be focused on standardization, integration with modern practices, and education and training. Future directions for Ayurvedic pest and disease management include enhanced scientific research, policy support, farmer training. Finally, it can conclude that Ayurveda pest and disease management offers a sustainable, eco-friendly alternative to conventional agriculture, requiring research, policy support, and education for global food security. These are my references, and thank you.
Okay, thank you very much, Dr. Kaushalya. Now, presentation is open for the discussion. I have a small clarification from you, Dr. Kaushalya. Can you give some example, the live example for use the pest and disease management through the crop rotation, how it's using in the Ayurveda and how it's interaction with the Ayurveda and that agriculture things?
There are a lot of remedies mentioned in Ayurveda for the pest management. There's a remedy by using arka milk, or we call it as vara, Abrus precatorius milk, and also Ayurveda mentioned about various fertilizers, organic fertilizers, which are made from sesame oil, meat, fish washing water, and also another recipe called panchagavya. These panchagavya made up from cow dung, cow milk, and cow's urine, curd, for the and used for the pest control, and also some of the some of pest management fertilizers include herbal materials also, Colombo, kaha, like that.
Okay, the Dasaparna Kasaya, Panchaparna Kasaya also same like that. No? Okay, thank you.
So I have one clarification. Do you have any idea the areas in Sri Lanka where this Vuksha Ayurveda used for the agriculture in the practical places?
Today I couldn't find about that because I go through the literature part.
Any literature
I couldn't find.
Did you find any literature from Sri Lanka? I think in that biodynamic farms, that areas you can find that Ayurveda we can combine with the agriculture. The practical purposes, if you are fine, I think there should be some places we are use this Ayurveda in the agriculture. Thank you.
Okay, thank you very much, Dr. Kaushalya, for the nice presentation. Now we are coming to the last presentation. Evaluation of different soil amendment and foliar spray on growth and yield performance of black gram. So the presenting author is Mr. V. Tilakshan. So I cordially invite to the Mr. Tilakshan to share their valuable finding with us.
Good morning everyone. I am Tilakshan Varadarajan from Department of Agronomy, Faculty of Agriculture, University of Jaffna. Today I am going to present my research title is Evaluation of Different Soil Amendments and Foliar Sprays on Growth and Yield Performance of Black Gram. Let's move to the introduction. Pulses are the second most significant source of diet for humans. They are good source of plant protein. Even though it contains high protein rich source of calcium and iron. They fix nitrogen, promote soil fertility, and even 80% of grown during a Maha season, and 70 to 80% of black gram is cultivated in Anuradhapura and Monia. These are my justifications. And my broad objective is to assess the effect of different organic soil amendments and foliar sprays on growth and yield performance of black gram variety Anuradha. And my specific objectives are to identify the effect of cow dung, poultry manure, and Prosophis uliflora as soil amendment on growth and yield, and to evaluate the impact of foliar sprays on growth and yield performance, and to assess the performance of Anuradha variety. And let's move to the materials and methodology. I did my research in the Integrated Farm and Training Center at Faculty of Agriculture, University of Jaffna. It's in Monia district. And the experiment was conducted in off-season. And these are my agronomic. And the main practice is amendment application. The three different soil amendments were chosen for my experiment, and those three are the three main plots. The main plots are A0, A1, A2. The A0 control only cow dung, and A1 is cow dung plus poultry manure, and A2 cow dung, poultry manure, and Prosophis juliflora. And these are my amendments. And then after amendment application, the seed of another variety were planted in the field with a spacing of 13 to 15 centimeter, and the seed requirement per plot is. And after the weed was hand weeded, by first weeding was done in 10 days after germination. And then the spraying. After the planting, the spraying was done 30 days after planting and 50 days after planting. These are my second treatment. And there are 5 treatments available for the foliar sprays. First one is Panchagavya, one is seaweed, and it's prepared from Catechizes aloe vera extract, and vermiwash. And that foliar sprays, the vermiwash was at as 50% and aloe vera was 50% and Panchagavya 4% of concentration and seaweed was 15% of concentration. And this is my experimental design, treatment, and their combination. The main plot contains 3 amendments and subplot contains 5 foliar sprays. And these are my treatment codes and combination. And this is my field layout with 3 replicates. And these are my readings I got while I'm doing the research. And after the planting of 55 days, the pots turning green color to black color. Then after the stem was cut and the whole plant was harvested, right, and then crushed and winnowed. Then after parameters was measured. The statistical analysis, split plot with 3 replicates was done. Let's move to the main part, the results and discussion. The first thing is growth parameters. The first one is plant height. When we look about the plant height, the impact of different soil amendments, the highest average height was observed in 7 weeks after planting, and it's in A2. It's cow dung, poultry manure, and Prosophis juliflora. Then the impact of the foliar spray, the higher height is observed in seaweed. and then the cumulative real number per plant is higher in A2 in the seed. In A2, highest root length was observed in seaweed, and then after the nodulation. The nodulation was higher in the A2, it's 50 averagely, and the lowest was in A0. We look about this foliar spray application. The highest number in foliar spray is seaweed foliar spray. The average flow number is higher in A2 in the seaweed extract. It's 6 weeks after plant. And let me look about the yield parameters. Average 100 seed weight is higher in soil amendment is A2 and in foliar spray is seaweed. And the average kernel per pot is higher in as a soil amendment it's A2 and in foliar spray is seaweed. The yield, total yield of it's in ton per hectare, it's higher in A2, it's 2.50 and higher in seaweed, it's 2.79. The combination effect of treatment on yield, the higher, even though the interaction was not significant, highest yield was obtained in A2, F2 treatment combination. And in Sri Lanka, the average black gram yield is 2.5 ton, but the recorded yield in this study is around 3 ton per hectare. It's 3.04 ton. The conclusion, considering all growth, yield and other parameters, seaweed extract was significantly performed well among all other treatments, even though in that amendment, the A2, cow dung, poultry manure and Prosopis uliflora is revealed that the results is higher. And the following thing is, in that seaweed extract after the Panchagavya, then aloe vera, and then vermiwash, and after finally the control. In that amendment, the first one is A2, second one is A1, it's cow dung plus poultry manure, 50%, 50%, and then finally the A note is control, it's cow dung 100%. And these results reveal that the growth and yield performance of black gram can be improved by organic amendments and foliar sprays without inorganic fertilizers and chemicals. And these are my suggestions. This is my important references. Thank you so much.
Okay, thank you very much, Mr. Tilakshan. Now, presentation is open for the discussion. I have a small clarification. So, based on your result, it revealed that the seaweed application, foliar application is give the promising result. So, what is the reason for that? When you compare,
when we compare that seaweed having that the important nutrients like micronutrients and macronutrients, it's available when it's applied to the leaves. Also, and it came from the literature, and then the seaweed having then the seaweed having that different with not only the micro and macronutrients. And other plant important nutrients are available.
did you check the NPK percentage of the seaweed application?
No, I didn't check, but in the literature I took.
Okay, so the better to check it, no? So then you
Yeah.
can get the idea about the NPK percentage. So then you can give a good result and good clarification regarding your result. Okay.
Yeah.
Right. Okay. Thank you.
Thank you.
Analyze the soil properties after application of soil amendments.
Sorry?
Did you analyze the soil properties after application of the soil amendments? That physical
No,
holding
I didn't analyze that.
I feel it's better if you are analyzing that properties. And other than that, I have a small clarification. Are there any special reason to select the Prosopis druciflora Sorry, Juliflora
Yeah.
plant especially.
Yeah. In a dry area, in a dry zone, the Prosopis Juliflora tree is it's very bad for the area because it's higher. It's accumulate higher number of water sources. Also, it's like a weed. So it's like a weed. So we don't have any benefits from that. They're having thorns in that stems, and also if that is an invasive species for us. So that's why I selected as the leaf. So we can reduce the weed as well as we can use as an amendment.
The objective to get a useful effect from that weed.
Yeah.
Thank you.
Thank you.
Right. Okay. Thank you very much, Mr. Tilaksan. So,
Thank you, madam.
okay, now we are going to conclude the crop production and protection technology session 3. So in this session, we discussed the 8 research finding, very valuable research finding in here. So I would like to thanks to the panel member, Ms. KG Katpayarachi and session coordinator, Ms. Chatura Sirivardhana for their valuable contribution to success this session. So dear all researchers, thank you very much for sharing your valuable findings with us. Okay, thank you.
Thank you so much.
Gratitude, our distinguished panel members, Ms. Supipi Navaratna and Ms. Kasuni Katapayarachi, to providing their valuable insight with the audience. And my dear authors, I'm very much thankful to you all to share your valuable research findings with NSATRS 2024. And I wish you all the best for your future endeavors. Thank you very much, and stay connected with our last technical session on sustainable technological advancement for sustainable agriculture. Thank you very much.
Good afternoon, everyone. It's my pleasure to welcome you all to the technical session under the theme of Technological Advancement for Sustainable Agriculture in Nsatrs 2024. Today, we have the privilege of being led by two esteemed experts in the field whose insight and expertise will be undoubtedly enriched for this session. I am honored to introduce our esteemed panel chair, Dr. Kumarage Tilanka Aryavansha, Senior Lecturer, Department of Agricultural Technology, University of Colombo, Sri Lanka. He holds a PhD in Bioresource Engineering from the University of Tsukuba, Japan. With extensive experience in agricultural system engineering, focusing on mechanization, automation, IoT, and AI, Dr. Aryavansha has made significant contribution to the sustainable agricultural practices and the integration of smart technologies. His research, which includes numerous peer-reviewed publications, focuses on sustainable agricultural practices and the integration of smart technology in agriculture. Dr. Aryavansha is proficient in programming, web development, and engineering design, and has received several awards and certification for his work. He is a member of the Sri Lankan Institute of Agriculture, the System Dynamic Society, and the National Science Foundation of Sri Lanka. And we are also privileged to have the Dr. Anushka Bandara as the panel member. Dr. Bandara is a lecturer in the Department of Agricultural Engineering and Environmental Technology, Faculty of Agriculture, University of Ruhuna. He holds a PhD in Regional Resource Environmental Engineering from the United Graduate School of Agricultural Sciences, Kagoshima University, Japan. His expertise in agricultural system modeling, greenhouse gas emissions, and energy and the biomass technology has been widely recognized and honored. So we look forward to engaging and enlightening the session with our distinguished panel. So dear sir, Dr. Tilanka Aryavansha and Dr. Anushka Bandara, I warmly welcome you to the session. And dear sir, this session is over to you.
Okay, good afternoon, all of you. Am I audible? It's clear? Right? Okay. So without wasting time, because we have to start at 2:50. Now we have 10 minutes past. So without wasting any time, so quickly we move to the session. So first, I would like to invite the first presenter, the manuscript number MS12, that the systematic literature review on the application of unmanned aerial vehicles, technology for paddy cultivation in Sri Lanka. The presenter is Mr. M.D. Madhushan. Okay, Madhushan, you can start.
Good morning. I am Damit Madhushan from University of Colombo, and today I will be presenting our systematic literature review on the applications of unmanned aerial vehicles technology for paddy cultivation in Sri Lanka. Rice is the staple food of Sri Lanka, making paddy cultivation a critical component of our agriculture industry. This study explores how unmanned aerial vehicles are used in agriculture, especially focused on the paddy cultivation in Sri Lanka. This technology offers potential benefits such as increased efficiency and improve the crop management. The main issues we address is the lack of advanced technological solutions in Sri Lankan paddy farming, which affect productivity and crop health. In the literature survey, we identify various applications of UAV in paddy farming across South Asian countries. For example, India and Bangladesh use UAVs for weed management and blood index assessment. Other countries like Nepal, Bhutan, and Pakistan also utilize UAV for similar purposes, enhancing the overall farming process. Our study has three main aims. To identify how new technological can be effectively implemented in Sri Lankan paddy field to improve the productivity and sustainability. We conduct a systematic literature review by analyzing extensive research papers and case study on UAV applications in agriculture. Is involved through search of the Google Scholar database and relevant publications from 2005 to 2024, resulting in 25 studies included in our qualitative synthesis. We use thematic analysis to analyze the contents of these selected papers. Our findings indicate that UAV can be used in various ways for crop monitoring, measuring parameters like leaf chlorophyll and nitrogen concentrations, plant height and leaf area and leaf moisture content. For weed detection and control mapping, weed density and spraying herbicide for efficiency fertilizer applications, ensuring uniform distributions for diseases detection, enabling to timely pesticide applications. For soil analyzing and erosions and fertility of the soil, for yield estimation, the normalized using the normalized difference vegetation index method, for irrigation management and seed spraying. A case study from our selection paper from our Sri Lanka. One study at the Rice Research and Development Institute in Bathrekoda used UAVs. They are used to identify the major weeds using multispectral images. And another one is the using UAVs for assessing leaf greenness and chlorophyll. And UAV evaluated from nitrogen level using RGB image and reflects heat maps. And finally, another case demonstrated UAV based multi-spectral imagery estimated rice yield accurately, supporting environmentally sustainable agriculture. However, they are challenged to their widespread adaptations of UAV in Sri Lanka. Technical limitations such as limited flight endurance and data processing capability, environmental factor like weather condition affecting UAV operation, regulatory issues including air-based restriction and license requirements, high cost and their need for specialized training. To overcome these challenges, we are recommended enhancing partnership between government, research institute and farmers, promoting innovations and knowledge sharing, increasing investment in UAV technology and data analyzing, developing comprehensive regulatory framework to ensuring safe and effectively UAV use. In conclusion, UAV holds significant potential for transforming paddy cultivation in Sri Lanka by providing high-resolution data for precision agriculture. While other countries have made strides in this area, Sri Lanka has yet to fully accept UAV technology. By addressing the identify challenges, we can harness their benefits of the UAV for sustainable agriculture. A reference to all the study review are available here for anyone interested in future reading. Thank you for your attention. I look forward to your questions and feedback.
Okay, thank you, Mr. Madhushan. So I think we have 3 minutes for discussion, no?
Yes. Yes.
Okay, so based on the study, so what is your recommendation? What is the things we have to implement first to like get the benefit from this technology for Sri Lanka? What is your recommendation?
These are the recommended. This is better than to traditional method means UAV is the better because pest controls and weed managements are weed management and distributions are to herbicides and high resolution real-time data that allow the persistent monitoring of crop conditions, detections of issues such as pests and diseases, and effective applications of input like fertilizers and
Okay,
pesticides.
it's okay. So that is the benefits we have. So I asked how we can implement these things based on your.
Yes, sir. We are recommended as our Sri Lanka is better to useful these applications to UAV unmanned aerial vehicles.
So how about the cost for this application of drones? Let's say for apply fertilizer for 1 acre. So what about the cost?
Yes, normally using high cost, but these are the future. These using our minimum cost designed some applications are designed these applications are we are minimizing the cost and applications fertilizer and other applications.
Okay, thank you, Bandar. I think you have questions
Yes,
now. Okay.
Manshan, thank you very much for your presentation. So I have a small question. So since you conducted thematic analysis, right?
Yes, sir.
Yes. So what is the thematic area that most researchers have focused on based on your outcomes? So that means if you categorize them into under several thematic areas, you may find this area will have this much of studies like that.
We have studied the applications, challenges used in Sri Lanka, UAV, unmanned aerial vehicles, and how to optimize the use in our Sri Lanka.
So any specific areas, I mean, be detection or like fertilization, any area currently Sri Lanka is mostly focused on?
Mostly focus in fertilizer applications,
Yes.
and
so okay, then based on your analysis, can you identify any thematic area that we need to have and we need to enhance our focus? So if you think if we enhance our research areas, if we extend our research areas to this area, it will be beneficial for us in future like that. Any idea?
yes, future is future. Our benefits may benefits of pest controls, weed detections, and likewise. Herbicides and herbicides applications, yield estimations, benefits of drones are used into paddy cultivations.
Okay, thank you. So
Okay. Thank you, because we have only 10 minutes, so we have to quickly move to the next presentation.
Okay,
so thank you, Madhushan, for this is interesting study, but you can continue this one because we can get this technology to the Sri Lanka to benefit of the agriculture. Okay.
Okay,
so
sir,
thank
of course.
you.
Thank you, sir. Thank you, sir.
Okay, so next presentation is the manuscript number MS16, the integrating remote sensing for crop management in Sri Lanka, opportunities and challenges. The presenter is Miss K. Ambalika. Am I right? That is the pronunciation? Ambalika.
Yes, sir.
Okay. So presentation, you can start presentation.
Good day to you all. Sri Lanka, once known as Granary of Asia, has now faced many challenges that slashed agricultural productivity. So, today we have come up with the opportunities and challenges to improve crop management in Sri Lanka through remote sensing. So, let's move into our topic without any further ado. Ancient Sri Lanka have become accustomed to an agri-centered lifestyle and brought forth a generation depending on agri-food, as it is rich in resources. Once an exporter of grains has now importing rice and other staples. Therefore, as an urgent embracement of new technology and innovations, remote sensing techniques aids in precise monitoring of health, growth and yield of crop, leading to optimized resource management. Next, let's move into the aim of our study. This study aims to access the potential of integrating remote sensing technologies in Sri Lanka's crop management by leveraging global outcomes and proposing strategies to overcome barriers and maximizing the gains for sustainable agricultural development. Next, let's move into the next. Here, we conducted a comprehensive literature review analyzing 20 peer-reviewed publications on successful global applications of remote sensing in agriculture, from which we extracted insights and opportunities for Sri Lanka and also identified specific barriers to technology adoption and proposed potential strategies to overcome these challenges. From our listed publications, we could able to come up with some applications of remote sensing globally. So here the usage of Landsat 8 images, object-based image analysis method, and UAV mounted cameras have been used to detect plantation crops, vegetation, and crop structural properties. In addition to that, UAV-based imaging were used to detect early pest infestations, specific wilting traits, water stress due to flooding and drought, and also combining synthetic aperture radar and optical imagery for early crop identification. In addition to that, combining SAR and optical sentinel data for phenological state prediction has also been done, and also real-time disease monitoring and early identification and alerting of crop diseases has also been done using this remote sensing technique. To further add, the usage of UAV-based multispectral imaging has been done to accurately study the pre-harvest rice yield, and also a semi-supervised system has been implemented for wheat mapping in sunflower crops, and also the UAVs have been used in the timely rice grain yield. So these are some of the applications of remote sensing that has been used worldwide. So despite these promising applications, when planning to implement these applications in our country, it is not easy. Our country has to overcome challenges such as maintaining high initial cost for considering both high initial expenditures and lower utility costs. Along with it comes the issues of infrastructure in rural locations, such as scarce internet connections and electrical power, which can be countered with broadening broadband coverages, and then lack of skilled laborers. And also there is no proper technical competence among farmers, and in addition to this, there is no proper policy guidelines or framework. So as addressing these challenges, we have also come up with some strategies like including state grants, international and local financial aids to help farmers, public-private partnerships, implementing broadening broadband coverages and developing solar panels or off-grid power sources, and also implementing workshops and training sessions on technological usages and data analysis, which could be essential to bridge the gap in technical competence, and also regulations should be put in place to facilitate advantageous technological promotion without violating farmers' privacy. And also implementing support services centers and helplines, introducing easy-to-use tools and software, and also implementing awareness campaigns and demonstrating plots and incentive programs for farmers can also be helpful. Hoping all these strategies would help in overcoming the mentioned challenges, we can conclude that by addressing these challenges and strategies, our country could come up with innovative solutions for sustainable agriculture. These are our key references, and that is the end of our presentation. Thank you.
Okay, thank you, America. So I have one question. So you mentioned that this is a remote application of remote sensing. So in Sri Lanka, we have a different kind of agricultural ecosystem. So different agriculture ecosystems, we use different technology. So based on your experience, your study, so you mentioned that there are a lot of challenges we have. So based on your study, so can you recommend which agriculture type, let's say farming system in Sri Lanka can apply this technology in recently? Did you get my point?
Yes,
so,
sir.
Yeah. Which agricultural system, maybe the plantation sector or paddy sector, which agricultural system is more suitable for apply this technology?
As the main production is rice, we can implement these systems in our paddy cultivation. And also, in order to increase our sugar production, we can also implement these in sugar plantations as well.
So in that case, what kind of productivity you are expecting? So as example, now without applying remote sensing, we are getting some yield, and we have some cost of let's say to produce 1 kilogram of rice, we have some cost, or produce 1 ton of the sugarcane, we have cost. So based on your idea, your study, can you give some rough value, how much of yield you can improve, or how much of cost you can reduce from this technology? Do you have any idea? Because when you are proposing some policy changes, so we have to do this kind of calculation.
So almost this by using this study, anyway, we will be able to identify the initial the damage stage or something. For example, we can identify the any disease initiations, or if the plant is going to get affected by any drought or flood stress due to any drought or flood stress. So in that case, we would be able to identify those things initially, and we can able to do we can come up with some ideas to prevent those at the early stages, even if it is a disease attack or something. So we would be able to identify them at the initial stage, and before.
I know that we can do that kind of thing. But when you proposing something, so after study, we are proposing the policymakers to implement in this technology is good, so we can improve the yield, or we can reduce the cost likewise. So my question is, how many percentage you can improve the productivity? It may be a yield, or it may be the cost reduction.
In both sides we can see, but almost I think we can increase our yield up to around 30 to 40%, and reduce the initial cost by at least 60%.
Okay, thank you.
Thank you.
Any question from Vandana?
Yes, so Ambika, and my concern is, how did you ensure the transparency in your study? I mean, did you follow any systematic literature review methodology, or like Prisma or something? Because so anyone can say this kind of thing without doing proper literature survey, or otherwise your conclusions may be tending to some biasness, right? So towards your conclusions. So how did you maintain the transparency? I mean, did you follow any systematic literature review methodology or
Yes,
something?
yes,
Yes.
we
So
reviewed
what are the
around
criteria?
we reviewed around 20 internationally
Yes.
peer-reviewed research journal papers, and those are with high citations. And so among those, we studied what are the most appropriate one that we can implement in our country.
So how did you do that? Do you follow any exclusion, inclusion criteria or something, or did you selected any reputed databases to obtain papers?
We use some reputed databases, as well as we did a simple basic search as well with high citation journals. We mainly referred with we referred with high journals with high citations, and then from that we included the one which would be more suitable for our country, and then we analyzed according to that.
Okay, thank you. Please better to consider such a method like Prisma or something in future. Thank you.
Thank you.
Okay, thank you, Amrita. So we will move next presentation quickly. So the next presentation is the manuscript number MS1818, automated detection of potato leaf blight disease severity using conventional neural network by Ms. Gunaratna. Okay, Ms. Gunaratna, you can start.
Good morning, everyone. I'm Shamika Madhushani, and I'm honored to have this opportunity to present my research to all of you today at 5th National Symposium Agro-Technology and Rural Sciences 2024. The use of machine learning in crop disease detection is a game changer for farmers. So in this presentation, I will be discussing how this technology is being used in potato-late blight disease severity assessment. As you can see on the screen, research title is Automated Detection of Potato-Late Blight Disease Severity Using Deep Convolutional Neural Network. First, I would like to give you an introduction about potato-late blight disease. This disease, caused by the pathogen Phytophthora infestans, causes significant yield loss in potato cultivations. Here we can clearly see the difference between healthy potato field placed by infected potato field. Let's move to the disease symptoms. Primary symptoms start from leaves. For example, blackish brown lesions on leaves and stem parts. Eventually, this disease can lead to the death of entire potato plants. Next, I will focus on research problem. Due to lack of adequate skills and knowledge, Sri Lankan potato farmers struggle to assess potato-late blight disease severity accurately. The severity assessment commonly evaluated through visual observations with the support of potato-late blight key. Okay, do you have any idea about severity level of this given potato leaf? Some people may say it's 20% infected, while others may say it's 40% or 50% infected. This means these visual observations are subjective and can vary from person to person. So these kind of subjective decisions in severity assessment lead to error conclusions and error treatments. So next we will see how we are going to overcome this issue. These CNN models are gaining popular due to their ability to obtain higher accuracy in object detection process. And also, these CNN models have the ability to automatically extract features directly from input leaf images to recognize different crop diseases. Okay, these are the objectives of this research. General objective is to investigate a quantitative method for assess potato-late blight disease severity accurately. The specific objectives are to test image module to detect potato-late blight severity and to quantify potato late fly severity by using deep CNN model. Then these are the major steps included in methodology. First step is sample collection. In here, disease leaf samples were collected from potato fields at Agricultural Research Center, Italia. Then, under pathogen identification, Phytophthora infestans was identified based on morphological features of their white-colored wool-type colony and of large, clear, lemon-shaped sporangia through light microscope. Then, next step, digital image acquisition and image processing. In here, disease leaves captured using a smartphone camera by focusing full leaf visibility. Then these captured leaf images were cropped using image software. Okay. Then next step is image labeling. In here, leaf images were labeled into 12 severity classes based on their severity level according to potato laid by key. Especially this was done using expert judgments. Then these are the major steps of developing CNN model. Input labeled images, then CNN model implementation, fine-tune the model, output and evaluation, and finally model testing with unlabeled images. Then I will focus on results and discussion part. If you look at these two images, two graphs, you can see accuracy of the model was increased by merging severity classes. And also this proposed CNN model increased its accuracy into 94% with the different image augmentation approaches. And this given confusion matrix method also being used to evaluate accuracy of categorization. Then in model prediction part, according to this 94% accuracy, model provided precise prediction. To further illustrate this point, here we can see the randomly tested late bite infected potato leaves, which are classified into their respective severity classes, severity class 0, class 1, class 3, and class 4. Then in the conclusion part, proposed model can accurately classify infected leaves into different severity classes, providing quick assessment of the disease. And this can help farmers to make informed decisions regarding severity level of potato late bite disease. And the future implementation of this study is to develop a smartphone-based mobile application for accurately determining the severity of potato late bite disease in real time. Also recommend the correct chemical dosage for managing potato late bite disease. And this is the end of my presentation. Thank you all for your kind attention.
Okay, thank you, Gunaratna. So I have one question. So how many hidden layers you have in this CNN model?
Actually, sir, in this model, there are 4 layers: convolutional, pooling, and fully connected layers. There are 2 pooling layers, and then fully connected layers.
All 4 layers.
Yes.
So do you use the computer or the high performance machine to do this evaluation?
With the support of with the support of one of my supervisor, he just helped develop this CNN model, and with his support, I was done this training and evaluation model prediction, these all parts.
Because I asked this question because you are going to apply this for a smartphone, but if you analyze it in the computer high performance machine, so then you have to consider about the number of hidden layers on because of computational power. Okay.
Yes,
so if you have any questions.
Yes. Thank you very much for the presentation, and interesting, and results are really good, I think. So,
thank you, sir.
So what are the image augmentation techniques that you have used to obtain such accuracy?
So I used image flipping, rotating, and image zooming. I used only these 3 augmentation techniques.
Okay. Then, so you are planning to develop a mobile application, right?
Yeah, yes, sir.
So in addition, I have a few one suggestion. So if you can incorporate some model emulation to your model, maybe you can reduce the computational burden when you applying for Android application or something. So just to think. Okay, thank you. That's all.
Thank you so much, sir.
Okay, so any question from the audience, because I can give 1 minute. Okay, if there's no question, so we will move to next presentation. Thank you for interesting presentation. Okay, so next presentation, the manuscript number MS-27, the molecular docking and dynamic simulation of OSBBX13 DNA complex interaction in Oryza sativa L by Ms. KGWW Bandara. Ms. Bandara, the presentation over to you.
Good morning. Today I'll be talking about molecular docking and dynamic simulations of OSBBX13 G-box complex interactions in rice. Most of the studies of plant genes have been carried out using the model plant Arabidopsis thaliana. Arabidopsis BBX21 is a transcription factor which has an ability to bind to G-box cis elements in the promoters. This gene plays key role in plant development and abiotic stress responses. Rice BBX13 is a close ortholog of Arabidopsis BBX21. Since main functions of orthologous genes are conserved during evolution and computational methods are extensively used to study DNA-protein interactions recently, the overall objective of this research was to predict the interactions and stability of OSBBX13 G-box complex using the molecular docking and dynamic simulation. Docking tool Haddock 2.4 server was validated by pre-docking protein G-box complex retrieved from protein data bank using Haddock 2.4 server. 3D structure for the OSBBX13 was predicted using ITASA and the 3D structure of the G-box DNA was retrieved from protein data bank. Molecular dynamic simulations was performed using GROMACS software for 200 nanoseconds. Analyzing protein G-box complex interactions after docking and superimposition of docked complex over reference structure revealed Haddock 2.4 server was a valid docking tool. Therefore, this Haddock server was used as the docking tool in this study. Here is the predicted 3D structure of the protein and the 3D structure of the G-box DNA. Molecular docking of G-box with OSBBX13 using Haddock server generated 13 clusters. Cluster 3 was selected as the best cluster. Molecular dynamic simulations was performed for cluster 3 complex using GROMACS software for 200 nanoseconds. The stability of the DNA protein complex was analyzed using GROMACS inbuilt tools, root mean square deviation, radius of gyration, solvent accessible surface area, and the hydrogen bond number. The RMSD profile of protein DNA complex shows that OSBBX13 G-box complex is stable after 10 nanoseconds in the simulation run. The radius of gyration remains constant after 10 nanoseconds in the simulation run, indicating that protein DNA complex is highly compact. The result of the solvent accessible surface area indicates that protein DNA complex is stable in the aqueous medium. Protein DNA complex formed hydrogen bonds in the range of 5 to 22 throughout the simulation process. Protein DNA complex is stabilized by both hydrogen and hydrophobic interactions. In this table, you can see the hydrogen and hydrophobic interactions formed in pre-MD and post-MD complexes. Here, you can see the two-dimensional and three-dimensional view of the protein DNA interactions after 200 nanoseconds MD simulation run. From the molecular dynamic simulations results, we can conclude that rice BBS13 can form stable complex with G-box DNA cis elements present in promoter regions of genes. The rice BBS13 G-box complex is stabilized by both hydrogen bonds and hydrophobic interactions. Thus, this study predicts that rice BBS13 can bind to G-box promoter sequence to form a stable complex. G-box regulates the transcriptional activity of light and abiotic stress responsive genes. Thus, OSBB13 could be a good candidate gene for improving crops for abiotic stress resistance in future. I would like to acknowledge the financial assistance given by the University of Colombo Research Grant, and thank you.
Okay, thank you, Banna. So, you did like a molecular dynamic analysis. So what is the purpose of major application of these things in agriculture?
These transcription factors have not been studied in rice. So to do the experimental method, we need the more, they are very expensive and time consuming. Therefore, before doing the experimental method, using computer methods, the interactions and protein DNA interactions can be predicted using these computational methods. So, because these are transcription factors and these transcription factors are involved in abiotic stress response pathways in rice, these genes can be used to improve the rice for abiotic stress resistance, like salinity resistance, drought resistance.
Drought resistance. That means you can use this information for the plant improvement programs, etc.
Yes,
right?
This is like a preliminary study.
Okay. So after you conducting your simulation, so the dynamic analysis, so how do you validate your result? How did you get some in? Is that correct, or that's like a validation?
The computational methods were validated, like docking methods were validated, and also there is a Arabidopsis gene called Arabidopsis BBX21. It can bind with G-box elements that is experimentally proven previously. So there are evidences. Yes, that gene can bind with the G-box cis element. The rice OSBBX13 is a close homologue of Arabidopsis BBX21. That's why we chose this gene to do a computational study. There are experimental evidences for the Arabidopsis gene.
Okay. Any question from Madam?
Yes, so I am not a specialist on this subject. However, now explain what are the challenges did you face during the molecular docking, and that means what is dynamic simulations that you have did. So what are the challenges you have faced during your experiment? How did you overcome those like that?
I am also not a not from a computation background. I am also have a molecular biology and biotechnology background. I learned a lot of things when doing this computational project. Sometimes I refer tutorials like when we do the molecular dynamic simulations, GrowMax, using the GrowMax, we have the GrowMax have the tutorials. So we refer those tutorials sometimes to troubleshoot the problems.
Okay, thank you.
Okay, so in your case, this is a collaborative work, no?
Yes.
Okay, thank you.
Thank you very much.
Thank you. Okay, so we will move to next presentation. MS 3838, utilization of aerial image for enhanced nitrogen decision support in paddy farming. A case study in Battalagoda, Sri Lanka by Miss CLIS Fonseca. Okay, Ms. Fonseca, you can start your presentation.
Dear sir, she informed that she will not be able to present due to the unavoidable reason. So
It is 48, no? MS 48?
Miss 38, Miss CL Fonseca.
So I got the 48, that's why
Sorry sir, it is MS
I
38.
MS 40, okay. Okay, so MS 38, so the Miss Fonseca not available, so we will skip that presentation. And now we will move to the next presentation, that is MS 48, and the robotics and artificial intelligence for crop protection, advantages, barriers, and research needs by DSP Ratnayaka. Okay, Ratnayaka, you can start presentation.
Good morning, everybody. I am Dumita Sanjay Bandaratnayaka from Industrial Engineering Training Institute, Katubedda, Mortal, Sri Lanka. My research title is Robotics and Artificial Intelligence for Crop Protection, Advantages, Barriers, and Research Needs. Okay, let's go to the introduction of my research. As per the predictions of World Food and Agriculture Organization, the global population will be increased by nearly 2 billion in 2050. It means we should work towards the feeding of day-to-day increasing population in the world. Increase of agricultural dates, unforeseeable climatic problems, and biotic stress are affecting for the creating of such type of situation. While 2040 of global crop loss is arising by insect pests and diseases. Minimum knowledge and expertise on crop protection create dependency of farmers on pesticide. It makes some negative impact on human health, biodiversity, and the environment. Therefore, awareing about the current technological advancement are needed. So then, artificial intelligence and robotics evolve from other techniques, generate cognitive solutions for the crop protection. So we should apply that technology for the enhancement of current agricultural practices. Okay, let's go to the objectives of my research. First one is identification of the advantages in the application of robotics and AI in crop protection. Second one is identification of the barriers in the approach. Third one is identification of the research needs to be enhanced, need to enhance the technology. Okay. Let's go to the methodology of my research. Basically, I used a qualitative methodology throughout my research. Both international and international literature were utilized for the creating of my report. So then, for the exploration of advantages, challenges, and research needs of the applications of robotics in crop protection, systematic literature review was conducted. Okay, Sometimes I used Google Scholars and some books and conference papers for the completion of my research. Let's go to the results and discussion of my research. First one is advantages. Robotics and AI are very recent technologies comprised with safety, preciseness, efficiency, and cost effectiveness. In here, functioning with sensors, cameras, and machine learning algorithms can be seen. So the utility in the pest and disease diagnosis, detectability of severity levels of damages, and being an alternative solution for chemical crop protection by reducing negative effects on the environment can be seen. Let's go to the barriers of the technology, barriers of the approach. Afflicting on climatic changes, imperfection and invasive pest and disease problems, requirement of high technological skills and low affordability of farmers describes the barriers on the technology. Okay, next thing is what are the research needs? Okay, what are the research needs on this approach? First one is collaboration of both public and private sector are needed. We should get the help of some scientists, some private company holders, and some researchers towards the enhancing of that approach. Next thing is we should conduct the social experiments on selected problems. Specific thing is solar and micro robots like modern technological creations can be utilized for the approach. I like to go to the conclusion. Robotics and AI-based crop protection approaches have the ability to make reincarnation of sustainable crop protection, reducing the harmful impacts on the environment, improved accuracy, efficiency, and cost effectiveness of the technology are pivotal among addressing of some challenges. Collaboration of both private and government sectors towards the new approaches on the technology aim to policy decision is worthy. There are a number of references. I use that references for the enhancement of that research. Okay, thank you very much for.
Yes, thank you, Ratnayaka. So, any question from audience, Bandar?
Okay, thank you, Mr. Ratnaik. And so, based on your findings, can you elaborate a little bit about what stage in Sri Lanka currently in regarding the application of robotics and artificial intelligence?
Actually, sir, the application is very low.
Yeah.
Yes. The thing is actually, sometimes some papers are telling that some approaches are there, but I think actually practicable applications are very low, sir. Because today I saw that some companies are going to promote the drone technology for the pesticide application of something. This is under Sri Lankan condition. I think this is the introducing stage of that technology under Sri Lankan conditions. Actually, the level is very low, sir.
So, what are your suggestions to overcome such things? I mean, more specific suggestions, right?
Actually, most important thing is people should be aware about the enhancement of their knowledge regarding that technology. I think this is not an isolated knowledge. We should combine the agriculture knowledge with engineering knowledge. So then we should get the partnership of both government and private sector relevant peoples. At the Sri Lankan conditions, in most occasions, most of the scientists are engaging with that technologies. But I think it is better to get the help of some sponsorship from the relevant parties, and otherwise, I think the government involvement should be needed.
Yes, so funding issues, right?
Yes, funding issues.
If we can, any suggestions like farming community introduction or something to overcome such financial issues? So, for particular cooperative society can buy some drone 1 or 2 like that and incorporate like that.
The thing is actually most of the when we are considering about any other countries with high technology, they prefer to donate their technology under for Sri Lanka like countries. I think we should discuss with that countries, and we should make some partnership programs under that category. I think these are my suggestions to enhance at that technology under Sri Lankan conditions.
Okay, thank you.
Thank you, sir.
Yes, sir, so I have one question. So
Yes.
based on your feature survey, so which countries perfectly use this new technology?
Actually, most of the Asia, especially Japan, Thailand, like countries, and especially in the USA, Canada, like countries are using that technology, sir.
So they use it effectively to reduce the cost of production? Because
yes, when we are considering about the high extent of then there are two types of agricultural lands, no, it can minimum extent and high extent. I think these technologies are effective for the high land extents.
Okay, so one question from this question. Sorry, because it's interesting for me. So you mentioned that when we expand the land sizes, so we can apply these technologies. But when we going into my idea, especially for the large field, we need bigger machines.
Yes,
but for when we lost robotics, how we overcome these things? Because robots are very small, so we cannot. We can incorporate robots for the bigger machine. But
Yes.
when you think about small robot application, so can we improve the productivity of the small farm using the robot?
Yes. I think actually the small farms are not practicable when we are considering about the profit coming from that farm. I think this technology is not effective. So in here, I am not discussing about, not only I'm discussing about pesticide application like that, the most important thing is diagnosing of some pest and disease problems. The thing is, if we are considering about the high extent of land, I think it is better to apply that technology because sometimes the detecting of various types of pathogen and insect attack can be done under very efficient manner. I think as an example, in we are using that without technology, we should see each one and make a time. No, I think it is better to use that technology for the enhancing of the detection technology like that. Not the detection technology, efficiency of the diagnosing of various diseases like that, sir.
Yeah, it is important because in Sri Lanka we have especially the indoor farming.
Yes.
The pest movement.
Yes.
Detection of pest movement is very important. So in that case, the detection is.
Yes, the thing is actually sometimes under Sri Lankan conditions we are treating or we are getting some treatment like we are treating the crops we are treating after the emerging of that problem. But I think this technology can overcome the more treatable conditions before emerging of the disease or any other insect pest damages, I think.
Okay, thank you, Mr. Naik.
Thank you very much, sir.
Okay.
Okay.
Okay, we will move to the next presentation. Manuscript number 54, MS54, Computer Vision in Livestock Disease Management, a review by Ms. G. Sibiha. Am I right? The pronunciation?
Welcome to everyone. I am Sibiha, here to present on my review on computer vision in livestock disease management. So let's go into the presentation. So this is the content of the presentation. So let's have an introduction. There is a historical evolution that has influenced the genetic trait of both livestock and agriculture in order to optimize the production and also to meet the human demands for food. So, the computer vision has been used widely in the livestock management and production, combining with artificial intelligence. So, there are several applications of computer vision used in livestock farming, such as monitoring animal welfare, animal health and behavioral studies of animals, morphological studies, and studies on the animal-based food processing. So this is the objective of the presentation. It is to optimize the application of computer vision in livestock management like in cattle, sheep, and pigs, and also to identify the advantages, challenges, and research gaps of the computer vision application. So according to the materials and methods, initially we collected the data with the literature search conducted in peer-reviewed articles. So about 278 peer-reviewed articles was used for this study, and we excluded several articles with lack of methodology and diseases of other animals and the techniques of other computer visions, and the studies with so some of them are infrared and thermal cameras, high quality mobile cameras, monochrome video cameras, kinetic depth cameras, 2D and 3D cameras, stereo version cameras are some of them. So this computer vision has so much of benefits, and it is one of the preferred method at present. So it is one of the contactless reading. So it also allows automatic measurements, and it is a non-invasive and non-intrusive method with more precise methods, and it also provides consistent measurements. So when focusing on the results and discussions, here we will discuss about the diseases identified in cattle. So IR camera is used in cattle in order to detect the early temperatures, and a study has showed a strong correlation between these. And this also has shown that a change in the body temperature is strongly associated with the changes in the rectal temperatures. So, and another camera is the infrared thermogram. So, it is used to detect the diagnosizations of FMD in vaccinated and non-vaccinated cattle, and also automated radio frequency identification. It is used to measure the highest temperature in the calf's eye socket using thermal imaging. So let's focus on the disease identification in sheep and pigs. So in sheep, the infrared thermograms are used. As in cattle, it is used to detect the early temperatures, and in pigs, IR camera is used to identify the infected and the healthy ones. So in computer vision, there are several limitations. So this computer vision has an initial high cost, and it also has a widespread application. Therefore, the high cost is high, and IRT is not disease specific, and several factors that can affect the accuracy of these computer vision cameras, like environmental conditions, body state, and cleanliness, and maintaining the sanitary conditions in the in the animal is a challenging task, and these cutoff temperatures that are being detected by these computer vision cameras will be affected by some factors like season, sex, age, and health status. So let's move to the conclusion. The thermal imaging is used widely in the livestock disease management, and we can use these thermal imaging cameras to analyze large number of samples. And these are the references. And thank you for giving me this wonderful opportunity. Thank you.
Okay, thank you. Mr. Bandara, any question from your side?
Is this again a literature review, right? So can you elaborate on main barriers to adaptation of, so my question is, what are the primary technical and infrastructure requirements? So need to implement this kind of technology. And if we adapt this kind of technology to a farmer or some local condition like Sri Lanka, so how do you have any suggestions them to consider when overcoming these initial challenges?
Having many advantages. For example, if we have to detect the diseases at the early stage, we can use this computer vision. And before the visual symptoms, using this cameras, we can detect the early detection of the diseases. Okay? But for, according to my suggestion, I would suggest to develop the computer vision using some AI technique. Therefore, we can overcome many challenges that are faced by the farmer. And also, we can diagnose or detect the diseases or the health state or monitor any of the characteristics of the animal in a large amount rather than one by one. Normally, we get the detect the diseases for each of the cat. For example, if we have a farm, we can detect the diseases one by one as a traditional method. But using this computer vision, we would diagnose the diseases for a large sample, and we can restrict the spreading of these diseases. So it would be an advantage, and the cost of production would be we can reduce the cost of production by preventing these diseases.
Okay, I have a question. So, are there any the machine vision application in Sri Lanka in the livestock sector?
Yes, so we are using IR cameras and IR thermography at present.
For detection temperature?
Yeah, detecting the cutoff temperatures and yeah.
So, other than that, what are the other usage of this machine version? So, other than like temperature, so what kind of other benefits we have?
We can monitor the even not only the live animals, we can also detect the qualities of the meat. Animal-based food production also.
Animal, sorry?
That means the dairy products like meat.
A dairy product.
Yeah.
So, so that means machine vision. Which kind of properties we are measuring that dairy product? You mentioned dairy products or the dairy related animals?
Dairy, that means dairy related animals as well as the meat products obtained from them.
Not dairy products. So, any other applications other than the temperature detection in Sri Lanka? So, are there any other applications? So, based on your literature review, what are the other applications in other countries? Machine vision application other than that temperature?
So we can measure the lameness, that means
Any disease detection?
yeah, this is detection of the lameness, that means
Lameness.
lameness. And
That is or the like.
Then we can also detect some bovine respiratory diseases and also
Can it be used to improve the quality of the animal products, maybe like meat or the meat quality, milk quality, etc.? So using machine vision.
yes, sir.
Okay. So then, what is your recommendation to Sri Lanka based on your literature review?
Actually, normally this computer vision does not have advanced technology to diagnose specific disease. So we can make some applications or develop a machinery by incorporating AI sensors as well as the IR cameras and like a software we can install any programs to that machine to diagnose a particular disease by analyzing the symptoms.
Okay. Okay. Right. Thank you, Ibiha. Sorry for wrong pronunciation. Okay. Thank you very much. We will move to our next presentation. MS55. UV-based multispectral imaging and the machine learning model for non-destructive prediction of sugarcane maturity by Mr. K.M.K.I. Ratnayaka.
So my research title is UV-based multispectral imaging and machine learning modeling for non-destructive prediction of sugarcane maturity. We all know sugarcane plant is belonging to family Fabaceae or Grassy family. It is used for production of commercial sugar and other byproducts like ethanol, paper, and bioenergy. According to the SARA industrial statistics in 2022, there are 78,000 metric tons of sugar production. But it is not enough for fulfill local sugar consumption. Therefore, the importance of increasing sugarcane productivity is the key. That's why new technologies are important. By implementing new technology, we can monitor real-time crop data and also real-time fertilizer applications. And also we can harvest crop at correct maturity stage. There are some challenges and key issues in sugar cultivation. Identify correct maturity stage for harvest. It is difficult to determine the optimal time to harvest sugarcane fields. Results in the potential for crushing the mature sugarcane. And using ground-based mixed values for maturity check. Mixed values helps measure sugar content to determine maturity. But small mixed samples cannot accurately represent the entire fields. It leads to decreased accuracy in assessing maturity across all areas. Due to that key issues, there are some impacts in industries. First one is it decreased sugar recovery. Due to the crushing of immature sugarcane, it leads to lower sugar yield. It affects overall efficiency and profitability of the factory. Then we propose the solutions by using real-time monitoring for correct mix sample values. It uses multispectral imaging-based machine learning technology. It's used to identify correct mix, pulp, and CCS values in large sugarcane fields. Then we develop a new remote sensing models. Create a model to accurately find sugar yield parameters over large areas. Then enhance precision in monitoring and decision making. So, general objective of this research is to develop and evaluate remote sensing methods for monitoring yield parameters and variability in sugarcane using UV-based multispectral images. There are three specific objectives of this research. This one is to identify the suitable vegetative index to detect Brix pulp and CCS status using multispectral UV images. And to develop Brix pulp and CCS variability maps, we selected vegetative index. Then to develop vegetative index based model, predict Brix pulp and CCS values using machine learning models. This is the process pipeline of my research. I will describe in next slide. The research study was carried out at the Lanka Sugar Company Private Limited, Kalata. We use two varieties which is mainly used in the industry. Then the sugarcane sample analysis was done at 10th, 11th, and 12-month-old crops. Two regions of interest were demarcated in reserve with 1 square meter size. Then sugarcane samples were taken and analyzed with pulp CCS according to the recommended method at lab condition. Daily flight mission was conducted on the same day of sugarcane sample collection. We used DJNL multispectral to capture canopy images. Then multispectral image map development was performed using the shapes of hair. Then QGIS software were used to develop 10 vegetation indices. These are the vegetative indices of this research. The machine learning part. Python and quantitative notebook was used to run machine learning algorithms. Future selection was done at most suitable vegetation indices were selected using Pearson correlation coefficients based correlation mathematics. Determine the accuracy was done using prediction of determination and group mean square. We use three machine learning models for this research. First one is linear regression, random forest support vector regression. Results and discussion. Total Dash Vegetative Index showed the strongest correlation with Pixel and CCS code, SL96 128 variety and SL92 5088. These are the machine learning results of the linear regression model. This is a machine learning result for support vector regression models. This is a machine learning results for rainforest models. Our findings demonstrated the superior results with the random forest model compared to similar study by Chia et al. 2020, which is utilized simple linear regression model. In the study, Chia et al. Reported that curve in radius has the highest R-square values of 12 and CCS irrespective of the sugarcane variety using the linear regression model. Finally, we conclude that these findings highlight the effectiveness of the random forest regression model in accurately predicting Brix-12 and CCS values for both varieties as evidenced by both higher R-square values and lower RMSE. These are the references of my research, and thank you very much.
Okay, Mr. Ratnayaka, this is interesting study. Again, so now can you use this model to predict the yield in the maturity without doing any brick, the traditional brick checking? Are you applying this technology now? Because there's a good accuracy based on your research. Ratnayaka, are you there? Not available.
Please join, sir. Please wait, sir. He will join.
Okay. I have only one presentation.
I would like to know any co-author present here. RAM Chandana, SWRAPI Shah, TJL Prasanna O, SHNP De Silva.
No co-author.
It seemed that not said. He had mentioned that that co-author will be joined, but not there, sir.
Okay, then So we have another one presentation, no?
Yes,
So
sir.
shall we move to that one? Then after that, after he join, so he's asked to.
Okay, sir. Thank you.
So then, yeah, then we will give the questions at the end of the next presentation. Is it okay?
Okay, sir.
Actually, that study is interesting. So because there are some results, so we need to know if it is applied in field. Okay. Okay. Then we will move to the last presentation. This last one. Oh yes, last.
Today, I will present my abstract on the topic of spatial temporal variability of Sri Lankan rainfall from 1990 to 2020. I am Gandhi. These are my contents. I'll start off my presentation with a brief introduction. Rainfall has been the primary source of water not only for agriculture but also for an array of industries. Rain-fed and irrigated agriculture play a significant role in the Sri Lankan exports, while many industries rely on electricity which is largely generated through hydropower in Sri Lanka. There is a temporal and spatial variation in the rainfall pattern all over the world, and there is no exception for the Sri Lankan context. The variation in the rainfall has paramount validity in the fields of agriculture in order to decide the crop calendars, and it is also essential to manage electricity supply and to decide the criteria to minimize the risk of floods and landslides. Sri Lanka consists of 3 climaterical regions, and rain-fed and irrigated agriculture practices are common in these regions. Specifically, rain-fed irrigation system in the dry zone is largely dependent on the rainfall. There are 103 major rivers open to coastal waters along the coastal belt, and those rivers bring a large volume of water to the adjacent coastal waters. Total discharge of these rivers vary with the rainfall, and thus it will largely be important for the entire dynamics of the coastal ecosystem. Under this context, a comprehensive study on rainfall variability of the Sri Lanka is essential, and the present study was designed to evaluate the rainfall variability of Sri Lanka over the last 3 decades. Therefore, the main objectives of the study were to analyze the spatial and temporal variation of Sri Lanka over the last 3 decades. Going on to my methodology. So to gain spatial variability of rainfall, 52 rainfall stations were selected to cover the whole geographical area of the country. Daily rainfall data from 1990 to 2020 was obtained from Department of Meteorology. Rainfall stations were selected to represent 3 climatical zones: dry, wet, and intermediate. This is my study area with the selected rainfall gauges. Initially, data screening was done to assure the quality of data. The procedure started with a rough screening of the data, then it was checked for any trend in the data using Spearman's trend correlation method. The stability of variance and the mean was assessed using F-test and T-test. Finally, the data was tested for any absence of persistence using serial correlation coefficient method. Then the missing data was filled using standard methods such as inverse distancing method, arithmetic mean method, normal ratio method, and linear regression method. Thereafter, using non-parametric man-candle trend analysis and sense slope estimator method, monthly, annual, and monsoonal trend for each station within the Sri Lanka was computed. Finally, to assess the interdecadal variability of rainfall, the mean annual data was calculated for the period of 1991 to 2000, 2001 to 2010, 2011 to 2020 using all the 52 stations. RGIS software was used to plot the spatial distribution of these mean decadal rainfall. And IDW method was used to interpolate the mean annual rainfall values at regular intervals. So going on to the results, screening of all data satisfied all the tests, and out of 52 stations,
The man-candle and sun-slope analysis found a significant increasing trend in the first intermonsoon in Alutnora, Kandalama and Nigambu. While Bibilla, Kanum Kenya, Colombo and Mount Kadwe exhibited a similar trend for southern southwest monsoon. These are the stations with significant trends across the Sri Lanka, and the upward and downward arrows indicate the increasing and decreasing trends respectively. Accordingly, a significant increasing trend in the second intermonsoon was only observed for Halboda, while Dakwari, Mascalia and Castle show significant decreasing trends. A significant change was not detected for the northeast monsoon except for Mascalia with a decreasing trend. The mean annual rainfall exhibited a clear variation during the last 3 decades, as shown in the figure. The mean annual rainfall of 3 climate zones during these 3 decades exhibits a large variation, where the dry zone received more rainfall during the period of 2011 to 2020 compared to 1991 to 2000. There were some areas with an annual mean rainfall of less than 900 mm during the period of 1991 to 2000, while those areas were missing as those areas received more rainfall in later 2 decades. To conclude my research, there are significant trends observed in some stations of Sri Lanka. Moreover, there is a noticeable spatial and temporal variation in rainfall over the 3 decades. Further, the areas with a rainfall of 900 to 1200 mm were expanding from 1990s to 2020. Our findings reveal that the rainfall of 3 climatic zones has largely varied during the last 30 years, expanding the rainfall towards certain dry zones. This information will probably be more important for the new demarcation of Sri Lankan climatic zones. These are my references. Thank you.
Okay, thank you, Nandi. Any question from Bandara? Dr. Bandara?
Yes, interesting finding, and that means we are going to change our climatic zones recently, right? So how this rainfall distribution changes in this rainfall distribution pattern, influence for the agriculture sector or maybe other sectors in Sri Lanka in future? I mean, like hydroelectricity generation, so such kind of things may be affect, right? So in that case, how this your findings will address those things?
Maybe so. If we can identify the trends of the rainfall properly, then we can efficiently manage our water resources, and also we can manage our irrigation demand according to our trends, and also the crop rotation patterns. Also, we can select certain crops. We can select it according to the specified rainfall patterns, and also if these rainfall trend also can be helpful for using to predict or forecast any pest or disease which are related to rainfalls, and which can finally be helpful for risk assessment for the farmers.
So, in addition, recently we observed that there is sudden rainfall happen for only localized into a small area. So after we move from that area, there's no rainfall. So completely different condition. So do you find such kind of things during the analysis?
Specifically not yet, sir, because all this data was analyzed for a period of a whole year, like yearly analysis. Even though we collected the daily rainfall data, the final analysis for the annual data. So if you're going for a certain analysis like that, then we have to go for a peak analysis or any trends within the daily data. So I think those by observing that day-to-day life, we can observe there is a certain change in this rainfall pattern. So going on or moving forward with a daily rainfall analysis, so we can get an idea about how these are changing within a certain area.
Okay, thank you.
Okay, that's interesting. So it means in Sri Lanka getting more wet than the before, no?
Yeah.
Yes.
So So in this case, so can you imagine how much of water we have now compared to the many years back? So that means we have to design all the things, So like drainage systems, because we are getting lot of rainfall now. We have same land area. So then we have to design everything based on that rainfall, like the agricultural area, land use patterns, everything is affected now.
Even our reservoir management can be efficiently increased also. So if we can harvest this rainwater and if we can minimize floods and if we can collect those water, then we can go forward to a very sustainable.
Again, in same time, there are some people saying that the temperature also increasing, no?
Yes.
It's not right.
Yes, there is a rise in temperature also.
So are there any relationship between these two based on your idea? Like increasing rainfall and also increasing temperature?
Maybe, sir, because maybe
RH difference.
Yeah,
RH difference.
Okay.
Thank you, sir.
So is this first time you mapped this thing in Sri Lanka?
No, there was a previously research done for this rainfall pattern for like 15 years and so, and the rainfall stations also very minimum. Like they have not considered 52 stations. It was around 30 stations. So I thought if we can increase the time period, and certainly we can observe these trends, the changes day to day life in our day to day life. I thought going forward with more rainfall stations with a long period of time can give an accurate trend analysis.
You use 52 rainfall stations?
52 rainfall stations.
So how many in Sri Lanka we have so far, all together? Maybe they are private
I'm
stations?
not exactly
I
sure,
don't
sir.
know.
I'm not exactly sure, but I selected 52 stations which had 30-year rainfall period, because most of the rainfall stations are missing 30-year rainfall data, and some of the stations are newly developed. So these were the only 52 stations were only few which had 30-year continuous rainfall data.
So how about incorporating satellite data or something like
Yes,
data?
we have done that as another part of the research for the eastern province, because in eastern province there is a very little amount of rainfall gauges. So what we have done as part of this research is we have incorporated satellite images, satellite rainfall data, and together with the rainfall stations which we have currently in eastern province, and we have created a machine learning model to forecast to forecast the future rainfall accurately, since we do not have rainfall gauges in certain areas. So for those areas, in order to predict or to get a accurate value for rainfall, we need to incorporate satellite rainfall data. But even though we cannot directly use those satellite rainfall data, at least we can take those satellite rainfall data, we can compare it with our rainfall gauges. Then using any machine learning model, we can go ahead with a prediction. That is also a part of this research going forward.
Okay. More question, no?
Okay, sir.
Okay, thank you, Subramaniam.
Thank you, sir.
Okay, so I think all the presentations completed except one. So how about the availability of Mr. Ratnaik?
Yes, sir.
Hello, sir.
Yes.
Hello, sir. I'm TLJ Prasanna. As a co-author, Mr. Ratnaik couldn't attend to the discussion, but I can participate in as a co-author.
Okay.
Also, I can respond to your questions and I can answer that.
Yes, my question was that that research has some good accuracy, the good result. So, do you apply this technology now in field to predict or detect the maturity?
Actually, no, sir. It's still in the experiment level, because we in the pellet sugar industry already having that sugarcane plots. Therefore, we are still trying to apply in the feed level. Now it's only experimental level.
So what kind of approach you have to apply this, like getting image, and are you developing any platform to analyze this?
Yes, we are taking the multispectral image using the multispectral drone. That spectral band content near infrared, red, green, and like that thing, 4 spectrum band, including the image. That multispectral image used for the creating maps. That map called orthomosaic RGB map. That map used for the already derived scientists or globally scientists derived the already vegetative indices. I think you are familiar with the NDVI, like example by NDVI index, like that indices. We calculate the 10 vegetative indices, including the NDVI. Then we get the NDVI indices, and we correlation with the bricks faulty and CCS value in the ground field level. Then we are check the correlation value. Then we are using the machine learning model and predict the bricks fault and CCS values.
Yeah, in NDVI, so we know that NDVI related with the infrared and radiation, no?
Yeah, that's the equation.
So then what is the relationship with maturity and this curve? Can you explain in the scientific correlation between that the NDVI value and the maturity? So normally if the plant is in stress conditions, so we can identify quickly using NDVI.
Yes,
so did you analyze each brand separately?
Yeah, in this this research, we are not focused on the NDVI. We calculate the 10 vegetarian indices, including the NDVI. We have to find the correct indices for the predict bricks fall value. Therefore,
you have found one, right?
Yeah, they have found one. Yeah,
found one. Yeah, we have found one, that the chlorophyll red edge. That's the indices name. Because then after our findings, we are search the previous literature. The global scientists already found the chlorophyll red edge is best correlation indices for the bricks fall, like that thing, yield parameters.
So in your case, you only get that indices available in that.
Yeah.
Or you created some indices for you.
Yes, sir. That's all indices are already derived by the global scientists, but sometimes we can calculate the different spectrums band using our image. But in this, we are using the already derived equation and indices in the global scientists.
Okay, did you try to relate some band with, let's say, the blue or red or any separate band with the maturity level? Did you try?
No, sir. No, actually, no, because we are following the previous literature. That's the reason for that, because we have to implement in the global research into the Sri Lanka, because there are no any research publication based on the bricks ball predict in the sugarcane level in Sri Lanka. That's the reason for that.
Yeah, if you have data, this is one of my recommendations. So if you can not only related to the previous literature, you can think about that relating some different the bandwidth with the maturity level. Yeah. So maybe something you can find new things. Okay.
Yeah, so thank you for the suggestion. Yeah, definitely we will try.
Dr. Bandar, any question?
So interesting results. And did you find any? Did you use any image processing techniques before calculating indexes, like removing the ground cover or something?
Yes, because reflectance value and disturbance value we are considered already, because we are taking at the middle quadrants in the plots. Then there are in this we are using the QGIS and Agisoft metadata Metashop software. Then we are avoid the shading. We are the shading condition in the image. Is that is we can remove any shadings and buffer conditions using the Agisoft software for the creating automosaic map.
Okay, so what will how are you are going to introduce this into stakeholders or the farmers? So
Yeah.
because now you are using Agisoft and QGIS, and maybe after processing the data, you will develop that ML method or something. So when it comes to the ground level, so they cannot do that. So that is why this will not practice everywhere. So can you develop any online platform or something to upload image and just get the output like that? Any
Yeah,
idea?
that's a real application questions. But in this, I already said that in is the research experiment level. But first thing is we have to find the drone. But drone are not available in the everywhere. We have to provide the rental service for drone, and we have to provide the extension service about that for the ground level farm. Firstly, we have to that. But before that, we have to do in the firstly application in the commercial sugarcane in our Perlatta sugar cultivation before the introduce the farmers.
Yes.
This is the initial level of this research. We are still progressing.
So what stages of sugarcane did you consider for the image acquisition?
Yeah, we are taking the 10, 11, 12 in the maturity stage.
Last stages.
Last stages, yeah, ripening stages.
Okay, thank you.
Okay, thank you. Thank you very much. So I think all the presentations completed. So today actually, before wind up the sessions, I would like to say there is a lot of information related to the agricultural technology. So especially we went through several presentations about the UA application for paddy cultivation, remote sensing for crop management, AI for potato blight detection, and robotics and AI in crop protection and computer vision, and also UAV machine learning for sugarcane maturity detection. Actually, these things are essential for the country as a research topic. These are very important, but in some researchers, some presentations here was like the basic literature reviews, but based on that, you can give a good recommendation. So, but some research, we got some general recommendations. So, therefore, if you are going for the continuing this study, so my recommendation is do a very systematic analysis and give the valuable output from this research. Also, there are some good research like AI-driven CNN. So actually, all of these are in basic level. So we have to use this technology in real field. So therefore, not only limited to that research area. So please try to think about how we apply this technology in the field level. So that is the important thing, because at the end of this research, at the end of any research, the expectation should be benefit for the society or the industry or agriculture sector. So therefore, always try to match your research with the society needs. Okay, so we had very good session. So wish you all the best for all the presenters. And thank you very much. So Dr. Bandara, if you need to say something.
Okay, thank you very much for the conference organizing committee for inviting us for the as a panelist for the NSATRS 2020-40. And today we have gone through nearly 8 presentations. So it is good that almost all of them are focused on emerging technologies around the world. So that means our scientific community, especially in Sri Lanka, that means it tells us that we are almost already in line with the global community, right? So that is a good thing. So another two things I want to mention that so sometimes we gone through several presentations basically about literature review. So So when we are doing this kind of stuff, so please ensure the transparency of your study. Otherwise, we can have some biasness, and so in that case, so your conclusion and everything maybe depend on biasness. So in that case, we have standard literature review methods. So try to follow those things, and some interesting studies that basically UEA based and AI method. So if as a suggestions, so of course we are scientific community and we are doing things. So however, when we can enhance the practicability of that by applying such kind of mobile technologies or Android application or something like that, so it will go to the ground level, and that will definitely benefit for the future of Sri Lankan agriculture sector. So please try to do that. And with that, I will wind up my talk, and wish you all the very best for your future endeavors. And I wish that you all will have a good strength to contribute to the betterment of agriculture engineering in future. And thank you very much, and have a nice day.
Yes, thank you very much. Keep in touch with us also.
Now we are come to the end of the technical session on technological advancement for sustainable agriculture. I would like to extend my heartfelt thanks to our session chair, Dr. Thilanka Aryavansha. Sir, your presence added immense value to the session. And also, I would like to extend my gratitude to our panel member, Dr. Anushka Bandara, for sharing your valuable thought with us. So thank you, dear sirs. Lastly, I would like to thank all the presenters who share in their research finding with us. And with this, we are come to the end of the Nsatrs 2024. So thank you all. And we are delighted to inform that we are published e-proceeding of the Nsatrs 2024 within today itself. So thank you all.
Thank you.